Hello.
Hello, sir.
Hello.
Welcome. Welcome, sir.
Thank you. Great.
देते ना हॉटस्पॉट नाही झूम ना प्रॉब्लेम ना ते टाकते त्याच्यात पासवर्ड
आहेत ना अजून 5 मिनिटं मी 5 मिनिटात देतो आहेत इथे
येस सर वी विल वेट फॉर अनदर फ्यू मिनिट्स कारण की मुलं पण येणार आहेत आणि अजून टीचर्स पण जॉईन होतील
Okay, I'm here. Just let me know when you want to start and I'll put on the video.
Sure, sir.
Hello. Hello. Should we get started? Hello.
Hello. Yes, sir. We are waiting for Himanshu sir. Just a minute. Just in a couple of minutes we'll start.
Or any of the students who were there when I came online? You can say hello to some of them.
Hello, Chirmule sir. This is Janhavi Bhagwat.
Hey, Janlavi, काय म्हणतेस?
मी बरी आहे. तुम्ही कसे आहात?
Majid. Bhagwat sir was my role model, and I am who I am because of him.
Yes, he used to talk about you a lot.
I know.
आठवण काढायची तुमची खूप.
Hmm.
He was so proud of you.
Thank you. I like I said, it was he was instrumental in my existence. If it wasn't for him and Karmarkar sir, I wouldn't have been doing what I'm doing. Yeah, there are a lot of people online. I'm just curious to see if we met the other day. Any of the students can put
Yes,
on your
we-
video and say hello. I'd love to see some of you. Is Ulfat around? Maybe Raza?
Hi, nice to see you again.
Nice seeing you all.
Hello.
Hello. Great seeing everyone.
Can you hear?
Yes.
Okay, thanks. Welcome.
Thank you. Whenever you're ready, we can start.
Good afternoon. Hello.
Hello.
Yes,
Good afternoon,
we can
sir.
start. Can you make Himanshu sir and Usha madam co-host?
Yes, I will just make them co-host.
So we are ready to start. We can start.
Good afternoon, everyone.
Good afternoon, sir.
Good afternoon, sir.
Good afternoon, Chidmule sir.
Hello.
Hello, sir.
Good afternoon, our honorable guest, Dr. Chidmure, Principal Sir, Usha Madam, our Director, teachers and friends. On behalf of Research Advisory Committee, we welcome you to Anusandhan 2022. With the support of Principal Madam, Principal Sir, and Madam Usha Madam. We RAC organizes various research-related activities. Anusandhan is an activity which is celebrating Science Day. Students of various streams present research papers which were published long ago and which still have an impact on their syllabus and everyday lives. The broad aim is to give exposure to the students to make them understand the way science and investigation was done in a era which we are very far from, and also to realize the impact and relevance of that work today. When students read those papers, they also understand the way communication used to happen long ago. There would be letters, personal communications of scientists, which today have a lot of impact. And this activity, we hope, develops a deeper perspective of how research, investigation, and communication was done in earlier times. So, with the introduction of our event and what we want to achieve, I request Principal Sir to motivate us with a few words. Himanshu Sir, please.
Very much, Devraj sir, and a very good afternoon to everyone. Respected Dr. Chirmule sir, Dr. Kiran Kolwar, my other teacher colleagues, and dear students. As Dr. Devraj has rightly put it, RAC of RJC has been a very innovative since its inception recently, and the activity of Anusandhan is one such feather in its cap, where students are motivated to go through the classical papers and present their own interpretation and how be a part of a journey of how science has evolved over the period. We are very fortunate to have week after week meet one of our alumni, a very wonderful human being, Dr. Chirmule, who addressed our PG students last week, and today he is as a chief guest for this Anusandhan activity. Dr. Chirmule sir, thank you very much for accepting our invitation and being here today. We look forward to interacting with you once again very much, and also give a guidance to our students and appreciate them in the initiative that they have participated in. Thank you, Kiran sir, for inviting me here. Thank you, Devraj sir. Thank you, everyone. And we look forward to a very productive hours lying ahead. Thank you.
Thank you, sir, for your motivation and support and guidance throughout. I request Director Principal Usha Madam to guide us with a few words. Madam, please.
So good afternoon, everyone. Respected Principal, Dr. Himanshu Davda, Chief Guest for today's Anusandhan session, Dr. Narendra Chirmule, the convener for RAC, Dr. Kiran Kolwankar, Dr. Devraj Pawar, teacher colleagues who are members of RAC, and all teacher colleagues who are motivated and have been mentors to the students for this Anusandhan 2022. At the outset, I congratulate each one of you on National Science Day and pay my respect to Dr. Satya Raman for giving name and fame to our country, and the Raman effect. I think at those times, there may not have been very sophisticated technology, but the zeal and the interest in basic science to achieve great heights. And every year, 28th February is celebrated as the National Science Day. If we had the regular working, students would have presented their project. But last year, when the pandemic struck, I congratulate the team of RAC members who came up with this very novel idea of making students read some classic papers, interpret it, and present it to all of us. Further, it also brought in a multidisciplinary approach because you cannot do science in solo, you have to come together. And science doesn't mean only people from science, it requires good communications, you need the language departments, you need philosophy, you need sociology, you need various other branches, humanities, commerce, everything. So this has given a platform for all our students as well as teachers to come together and listen to each other. Today's chief guest will be formally introduced, but I think we could not have thought of a better person for this event because he believes in innovations, and I'm very sure he's going to share a story from the classic paper. So with this, I congratulate the entire team of RAC for bringing this Anusandhan for all of us. And I'm very sure students will also benefit by learning from each other. So thank you very much. I look forward to hearing to the section. Thank you.
Thank you very much, madam. I now request Dr. Rachna madam to introduce our honorable guest so that we can begin today's events. Rachna madam, please.
Yeah, good afternoon, everyone. I welcome Dr. Chirmule for this event, which is organized on the occasion of National Science Day. I would like to introduce him. Dr. Chirmule is the co-founder and CEO of Symphony Tech Biologics, a data analytics company focused on engineering solutions to biology. As former head of R&D at Biocon and in leadership positions in Amgen and Merck vaccines, has contributed to clinical development of vaccines and biopharmaceuticals. He did his bachelor's and master's from Jhunjhunwala College and a PhD from Cancer Research Institute, Mumbai, postdoctoral studies on pathogenesis of AIDS from Cornell University Medical College, North Shore Hospital, New York, teaching and research as assistant professor in gene therapy at University of Pennsylvania, Philadelphia. Dr. Chirmule is on the NIH Advisory Committee for HIV vaccines. He is a TEDx speaker and recently has published a book, Good Genes Gone Bad, by Penguin Press, both of which describe lessons learned from colossal failures in drug development. Sir, we are looking forward to hearing you talk on vaccines, innovations that transform human health.
Thank you. Thank you, Rachna. And thank you everyone for inviting me to this Science Day celebration and making me part of your exercise. So I love the idea of looking at historical papers and as a concept to understand biology. So let me share my slide deck. Somebody has to unshare their slides before I can. Yeah, I can do it. Can you? Yeah, I think you can see my slides now. Okay, so what I'm going to speak on is maybe my perspective of vaccine biology and how it has transformed human health, but also tell you a little bit more of the mechanism of immune memory. Now, I've taken the liberty to have a little bit more complicated topic to be discussed or to be presented, but I wanted to not only talk about the historic papers, but I also wanted to talk about the more recent papers. Even these recent papers that I'm presenting are at least 10 years old, so they are still a little historic. So I'm going to preface my presentation by talking about immune memory. So I like to show this picture because I love the spaces between science and art, for example. And this is Salvador Dali's painting, which is defined as persistence of memory. It's a very beautiful painting, and it sort of reminds us of time and memory. So that's the painting. the first time a vaccine was thought to work, or it wasn't called vaccine at that time, but in 430 BC, it was already observed that whenever a person got infected with the plague and did not die, that person was not infected again. So there was this notion that something allows you to develop immunity against reinfections. The paper I was going to present in detail was this paper, which is a historic paper written in 1846 by Peter Panem. And it's a very interesting paper. I will post it later on for all of you to read. It's a large paper. It's probably like 50 pages long and maybe even 100 pages long. And it talks about how infected people systematically don't get infected again. So what is the paper about? So the paper is about basically vaccines before natural vaccination. The study was done in Faroe Islands, which is on the right side. You can see Faroe Islands on the north of the British Isles. You can see Faroe is there. And it's an island where a very small number of people lived. But when Peter Panem did this study in 1846, he found that in 1781, there was a measles outbreak. For 65 years after that, there was no measles. In 1846, 75% of the population got infected. And he looked at two groups, an experimental group and a control group. And he said, in the experimental group, of all the aged people living who had measles in 1781, none of them were infected again. And of the control group of the aged people who did not have measles in 71, all were attacked. So this is in 1846, another example of population studies, very systematic population studies, which have proved that infectious agents can cause protective immunity. This is at a time when very little of the technology of antibodies were not known, nothing about the immune system was known, microscopes weren't really discovered at this point. So it was very interesting to make these natural experiment observations. Of course, now we know that when you get a vaccine or you get infected with a particular virus or bacteria, you generate a primary and a secondary immune response. And now, because of COVID, we know much more. सब लोग पूछते हैं second shot मिला, तो second shot मिलने से क्या होता है? So when you get the second shot, your threshold, the dotted line at the bottom, immune response that you measure, you need to get a response that is higher than the threshold so that you don't get infected. If the antibody response or the immune response goes below the threshold, then there's a high likelihood of getting infected. And so determining what this level of threshold is against which you will get protected against disease is one of the holy grails of vaccinology. So, sorry about the sound. So the question is, what is the cellular and molecular basis of this accelerated response to cellular immunity, to vaccinations? And are memory cells distinct from effector cells? And what are the lineage of memory cells? These are fundamental questions in basic immunology. And then one very important question was, is specific antigen necessary for maintaining memory? So in the 1990s, this was a question which was very difficult to answer. So in order to answer that question, I'm going to show a very elegant experiment that was done. Just to preface that, when an antigen stimulates the B cells get activated, and activated B cells then proliferate. They undergo class switching where they make IgG and IgM, and then they also become memory B cells. And many factors are involved in activation of these antigens. Klaus Rajewski in Frankfurt developed this, who's a very famous immunologist. He developed these what are called conditional knockout mice based on Cre-Lox recombination. And here is where it starts getting a little complicated, but I think you will understand, hopefully, of what Klaus Rajewski was trying to do with his group. So let me explain to you what this Cre-Lox system is. So the system contains of an enzyme called Cre-recombinase. Just hold that thought. Recombinase is an enzyme. And of course, enzyme is encoded by a gene of that enzyme. Now, this recombinase enzyme recognizes DNA. Recombinase means what? It is a DNA cutting enzyme. Where does it cut in the chromosome? It cuts in the chromosome when it recognizes something in the chromosome. So what does it recognize? It recognizes something called LOX sites. So what does that mean? So if you look in the middle, the recombinase enzyme, which I just told you, Cre-recombinase, will recognize anything that has the LOX site. So LOX site is a DNA sequence, which you flank the DNA that you want, the gene that you want to regulate. So this is VDJ is the antibody molecule for nucleoprotein. This is a protein against an antibody, the antibody molecule to a particular B cell. So you can imagine this is a B cell that expresses that antibody for nucleoprotein. So what Klaus's group did, they put these LOX sites on the 5' and 3' sides of the VDJ sequence. And when they put this sequence on the side, Cre-recombinase is able to recognize the LOX site and cut that portion in between. As you can see here, once you add recombinase, that middle portion goes away, and this B cell, which is making antibody to nucleoprotein, now doesn't make antibody anymore, right? So what does this mean? How does this experiment work? So you have a B cell in this mouse, which is expressing this antigen, pink antibody. This is that VDJ sequence, which is the antibody molecule. These are the LOX sites, right? The Cre is presented to this mouse, and by activating the Cre enzyme in that mouse, when that Cre enzyme is activated, and I'll do this one more time, this is the antibody producing B cell. When Cre is activated, that X gene is deleted on that allele of that chromosome. And interestingly, as soon as that allele is shut off, the homologous allele starts making the antibody to another antibody, which is called antibody Y. And now this B cell starts making the blue colored antibody. So just go back again. Antibody making B cell making the pink antibody, treated with Cre-recombinase. That antibody goes away, and this same B cell start making another antibody. This doesn't happen in nature. One B cell always makes one antibody, but in this artificial system, they were able to make that B cell make another antibody. Why is that important? That is important because only when an antibody is presented on the cell surface of this B cell will this B cell receive signal from that antigen to live a long life. By deleting the antibody, the original antibody, this group was able to ask the question, does the B cell survive for a very long time, even if it doesn't have the antibody for it to be activated? And the long story short, this paper was published in 2000, 20 years ago. Memory B cells activated with antigen X survive for the life of the mouse in the absence of antigen X. So antigen is not required for B cells to survive forever. So this was a fundamental finding which was done by this elegant set of experiments. So this becomes very relevant in COVID times, and a lot of things can be discussed on this, but I'll move on because of interest of time. This finding has been very fundamental in understanding basic immunology. We have lots of vaccines against many different pathogens listed here. And today we are talking in COVID, what is that level of antibody, that threshold? What is that level of antibody that it can elicit protection? For all these viruses and bacteria, the level of protective antibody that is shown here in the right column has been established after many years of breakthrough studies. It takes decades to figure out what the concentration of the antibody that really is inducing protection. That takes a very long time to discover. But this is what we know for all these other viruses. The one for COVID, hopefully we will know in a couple of years. Now, Maurice Hilleman is a very famous scientist. He was a very famous scientist. He is credited with saving more lives than any other medical scientist in the century, because under his oversight at Merck, vaccines for measles, mumps, hepatitis A, hepatitis B, chickenpox, meningitis, pneumonia, and influenza have saved millions of lives from death, right? So, and he comes from a very small town in the US called Miles City, went to Chicago, and then he was at Merck for 48 years. I like to give these examples of these famous people because all of these people's history tells you a little bit about what you yourself can achieve in terms of in your own goals. I'll tell you another B cell story, and then I'll tell you another T cell story, and then we'll stop. Long-term memory B cells, when you generate memory B cells, they don't circulate in our blood. So if you take blood and you look for memory B cells, they're rarely there. What Rafi Ahmad and his group at Emory University discovered in a very elegant set of experiments, that memory B cells actually they reside in the bone marrow. And Rafi is from Osmania University, went to Idaho, spent time at Harvard, Scripps, and UCLA, and he's been at Emory for a very long time. And relevant to this, for example, just look at the bottom picture where I've marked in red. Now, based on these understandings of long-term memory B cells, we are able to define people who are vaccinated with COVID, how long their memory B cells can last, and how long is the vaccine eliciting protection. So these are very, very important studies to study the biology of B cells. Let's switch gears and go understand what T cells do. T cells, like B cells, are also induced by antigen stimulation, but unlike B cells, which express antibody, where the antigen binds directly to the antigen, T cells can recognize antigen only when it is presented by MHC molecules. And what this slide summarizes a lot of data that each virus, in this case, HIV, cytomegalovirus, Epstein-Barr virus, herpes simplex virus, have a unique pattern of how T cells are activated in a time kinetic manner. So if I add COVID to this, it'll be even more complicated. Now, another Cree-Lox story. Now that you know how Cree-Lox works, let me show you another very elegant study to study what this group did from David Baltimore's group, is they tried to understand how do you look for T cells when you need very complicated methods for measuring T cells. So what they did is they took a gene, they took the Cree gene and made a mouse with that. And they made a very complicated gene pattern. What they did is this is a reporter gene, phospholipase enzyme. What this enzyme is, just think about this blue box as a reporter enzyme, which means this gene encodes for something that makes the cell turn blue. It's a reporter gene. So when the gene is expressed, the cell will turn blue. And remember the lock site, right? The lock site is the site where the recombinase Cree enzyme can act. Now, what this group did, Joshi Jacob and David Baltimore did, is they treated this mouse, which expresses this phospholipase reporter, because it has a stop codon. Around the stop codon, they put the lock site. So when the lock sites are present, this cell is not expressing blue. Because it's a stop codon, this cell will not be blue. But when they treat it with Cree enzyme, that middle portion will be gone, the stop codon will be deleted, and suddenly this cell will start becoming blue. And since the Cree is controlled by a promoter, which is a gene to the 5' of the Cree gene, it is actually only activated when activated T cells emerge as a result of vaccination. So when you get vaccination, activated T cells will emerge. When activated T cells will emerge, Cre will be expressed by those activated T cells. That Cre will delete the stop codon and the cells will turn blue. And this is shown beautifully in the slide. So when you see non-vaccinated mice, you see no blue cells. PLAP is the blue cells. And when you vaccinate, you can see all the cells that are the T cells that are activated now are turning blue. You can also see them in the germinal center. So this beautiful, elegant way molecularly engineered cells are allowing us to study memory T cells and memory B cells in a very, very methodical way. These are the kind of experiments that academic institutions do, right? And I'll end by just showing you there are lots of vaccines that we have, I showed you, but there are lots of pathogens for which we don't have vaccines today, including tuberculosis. Recently, there's been a new vaccine for malaria, which is not very effective, but it's at least 30% effective. And we have no vaccine for HIV. And obviously, we have a vaccine for SARS-CoV-2. So ultimately, the goal of the vaccine is to identify correlates of immunity to different pathogens and understand how these responses can be sustained, memory cells. That is critical for developing vaccination and long-term immune memory. So I chose this topic because of the importance of vaccines today. And as a biotechnology and a science group that you all are part of, it is important to look at very deep scientific topics. I know this topic may have been quite difficult for some of you to understand, but let me stop here and maybe take any questions. So thank you very much for inviting me. I'll stop and unshare.
Thank you very much, sir, for this talk. You would like questions from the students, teachers? You're allowed to unmute and ask the question.
And unmute and ask the question or type in the chat box.
Don't be afraid, ask any question. It was a very difficult topic.
So maybe by the time people are typing in, may I ask some layman type question?
Please, please do.
Also for COVID, how do we know when memory T cells, up to what time they saturate?
So very good question, Devraj. There's no such thing as a simple question. These questions are very difficult. So for COVID, what happens is we are looking at multiple things, right? We are looking at different vaccines being given at different time points. Just a second. What happens is the vaccine is able to induce a memory cell response upon one vaccination. Memory cells increases after 2 vaccination, and the memory cells will increase further after 3 vaccination. And typically what happens is the level of memory, today we are measuring it by neutralizing antibodies. There are very few studies where they look at T cell responses for memory. So the short answer to your question is each vaccine induces different levels of memory and different times. Like for example, when you take hepatitis B vaccine, you get 1 shot at birth, 2nd shot at 6 months, and 3rd shot at 1 year. When you take measles, mumps, rubella vaccines, you take the 1st shot at 1 year, the 2nd shot at 2 years, and 3rd shot at 5 years. So every vaccine has a different regimen of inducing memory. Because COVID is such an important or such a devastating disease, we are just taking a chance and hope that we generate memory in this short time frame. That's the short answer, Devraj.
Adwaita Singh has a question. Adwaita, you can unmute yourself and ask the question.
Hello, sir. I wanted to ask for the people who suffer from autoimmune disorders. So, in those people, how this T cells and memory cells function?
Oh, very good question. So the difference between the nature of the immune response to a virus and the nature of the immune response to autoimmune disease is actually the same. Means the nature of the immune response. What happens in autoimmune disease is that these immune cells that are reacting, they are reacting to a self-antigen. So the recognition machinery has become faulty. There's something that allows these cells that should not respond to self-antigen that start becoming self. The short, simple answer to that is once you get activation of, once you get an antigen, whether it's a viral antigen or a tumor antigen, in fact, tumor antigen or a autoimmune antigen, the nature of the immune response will still be the same. The cell will still secrete interferon gamma. The cell will still upregulate molecules like CD20, and the cells will still proliferate and express genes. So immune response is the same. Antigens are all different. Okay.
Thank you, sir. We have a couple of more questions in the chat box. If
Okay,
you can
great.
access the chat box, or I can read them.
Yeah, can you please read them? Thank you.
Yeah. There is a question by Tanya. In the nude mice cells, Krylov system, only one allele is acted on, but not on the other, on the homologous chromosome. Why so?
Very good question. Very good question. So we might require a little bit more discussion on that, but the simple answer is in molecular biology and in molecular understanding of our Krylov system, when we make these genes, when we express these LOX genes and Cre genes, we're putting them in the embryonic stem cell of the mouse. So we put them in the embryonic stem cell of the mouse. It is basically the fertilized zygote, right? When the fertilized zygote has the gene and the mouse forms from that fertilized zygote, all the cells of that mouse express that particular gene. And when you insert the gene into that particular chromosome, you're just taking a chance and hoping that the gene goes into the right place. You don't know exactly where it is going in the chromosome. So there's a little bit of a chance molecule. You transfect the cells and the gene goes in and it goes into the chromosome. At that time, unless there's site-directed insertional mutagenesis, it goes anywhere. So our chromosomes have a mechanism called inactivation of the other allele. Most of our genes, not all, but most of our genes, when one protein is expressed by one allele, automatically the other allele is shut off. And the mechanisms of why only one allele is on and other allele is off for various proteins is unknown to me. I'm sure molecular biologists know it, but that happens. But there are some proteins where both allele express the protein. But for the majority of the proteins, one allele expresses it. So that's the short answer.
Thank you very much, sir.
Thank you, sir.
Dr. Meher has a question. Why do vaccinated people get infected with COVID? Are the memory cells getting deactivated after some duration?
Yeah, very good and insightful question. So a couple of answers I'll give you. We don't know the full answer today. The couple of answers are, when we get vaccinated, today we are getting intramuscular vaccinations, right? We go and get the shot in the intramuscular route. When we get intramuscular vaccination, there should be enough antibody that induces high level of antibodies in the nose and the mouth. Generally, when you get intramuscular injection, you get a good systemic immune response, but not necessarily in the nose and the mouth. So most likely, in my opinion, what is happening is vaccination does not induce high level of antibodies in the nose and mouth. And that's why even though you're vaccinated, you get infected, but you have enough antibody levels so that antibody levels, systemic antibody levels, prevent severe infection. So that's the short answer. And as far as the second question was, are the memory cells decreasing? There are some reports to suggest that the memory cells are decreasing. The mechanisms are not clear, but we'll have to wait in time. It's not even been a year. So we'll have to wait and see if that is true, or if there's if the virus uses some mechanism to reduce the memory cells or not. It's not clear.
That's great to think that the virus can react and fight for its survival.
Yeah, I
Again,
mean, actually, I keep saying this, right? Humans are only 2 million years old. Viruses are 300 billion
they
years
are
old. हमारे से बड़े
trillion years.
बहुत बुजुर्ग हैं हमारे.
Yes, they have survived so long,
They know how to
multiples of the
survive.
epochs that we have existed. It's amazing
Exactly.
to think that they will fight us back. That's amazing.
Of course.
There are more questions. What are the
Please.
ingredients in COVID-19 vaccine? Preeti Yadav has this question. Ask to
Yeah.
her.
So, I mean, every vaccine has multiple components. The main component of most vaccines is the spike protein itself. Either it's the protein or the DNA or the mRNA, or the spike protein is expressed in an adenoviral vector. So the spike protein is majority of the antigens. Covaxin has the entire viral protein as the heat killed or killed virus by. So that is the antigen itself. Then to stabilize the antigen, there are many formulation compounds. So there are a lot of excipients that are added into the buffer, like a buffer, which are added into the vaccine, so that the vaccine doesn't degrade quickly, it remains stable, etc. In mRNA vaccines, a lipoprotein or glycolipoproteins are used to stabilize the mRNA from getting degraded quickly. So those are the major components of the vaccine.
Thank you, sir. Questions moving towards periphery. How to convince laypersons, those who are not ready for getting themselves vaccinated? Dr. Sharmila Jajodia has the question.
Political question, right?
Yes, from
There
the
is a.
periphery.
So how to answer this question? Let me tell you a story about this question, right? So in my opinion, there are probably like 3 levels of people who don't take vaccines. Three reasons why people don't take vaccines. There could be more, but if it doesn't take vaccines, there are probably 3 categories of people. The first category of people, I would say, are the real, they believe in the data, they have the data or they have seen some data, and they say, oh, the vaccine can cause these side effects, and therefore I will not take the vaccine. I don't want the side effects, and I don't want to, that is one group of people. The third group of people, I'll come to the second later. The third group of people are the people who have no data, but they don't care. तुम कुछ भी बोलो मैं नहीं लूंगा. वो मैं लूंगा ही नहीं, बस मेरे किसी ने बताया कि कुछ नहीं लेना चाहिए तो नहीं ले रहा हूँ. So those are the extreme groups of people, right? And in the middle are somewhere in between. So let me tell you the story for the last group of people who will say that I won't take the vaccine no matter what I tell you. I don't want to talk to those people, right? Because they've already made up their mind for whatever reason. It could be religious, it could be practical, it could be many reasons, right? I feel like I don't want to spend my energy to those people. They've already made up their mind, and I'm trying to, my return of investment should be high, right? So I believe in vaccines. So I'll focus on the people who I think I may be able to convince. So for this, I'll tell you a story. So I show you a friend of mine, and he's helped me quite a bit in terms of understanding vaccinology. Paul Offit, he's one of the world's most renowned vaccinologists. He's in the University of Pennsylvania. He tells a story. He's a clinician, and a patient comes into a little baby comes into his office to get vaccinated, a regular vaccine, not COVID, just regular measles, mumps, rubella kind of vaccine, right? And the mother is holding the child. Mother is holding the child, the vaccine, and he takes a syringe. He fills the syringe with the vaccine, right? He fills the syringe with the vaccine, and air bubble or nikal ke, he's getting ready to inject the baby with that vaccine. Just about to give the vaccine, not given yet, not about to give the vaccine, and the baby gets a seizure. Okay. Vaccine not given yet. If that baby got a seizure 1 minute later after giving the vaccine, no matter what you do to that you cannot convince that mother that it wasn't the vaccine. So this is the first group of people who have actually seen vaccine diya hai, कुछ हो गया, मैंने देखा है, मतलब that person will say मैंने देखा है, वैक्सीन दिया कुछ हो गया. Those people have their own data to say that vaccination is not safe. So those people are, the real data believers. We'll have to show them a lot of data, that the data that they have may be an anomaly or an outlier. And that's why maybe we may be able to convince them, because they are a data-driven group of people. If you show them enough data, I think you might be able to convert them. The next group of people are people who are sort of always oscillating between not taking the vaccine, or maybe I'll take the vaccine, and they're oscillating from one side to the other. And those group of people, you have to be persistent. You have to constantly give the message that the vaccine will work. And we have enough data generated on hundreds of vaccines given for hundreds of years, 100 years at least, that shows that the risk-benefit ratio of the vaccine far exceeds anything that the pathogen can induce. So those are the group of people you have to persistently provide data. So I know a little bit of a long answer, but that's my thinking about how to convince people who don't take vaccines.
Thank you, sir. This has been on everybody's mind. Within our close groups, we find, even within the family, we find this kind of opinions. So thank you very much.
So one more thing, Devraj, I'll tell you, which I think is very useful, maybe a useful methodology of telling people that I have started using, that there is no drug, there is no vaccine that is 100% effective, and there is no drug and no vaccine that is 100% safe. This does not exist. हर एक ड्रग का कोई साइड इफेक्ट तो होता ही है, राइट? एंड हर एक ड्रग 100% तो काम करता नहीं है. तो वैक्सीन का भी वही है. अगर वैक्सीन ले लो, हजारों जनों में इट मे वर्क, बट तुम्हारे इसमें नहीं वर्क करेगा. हो सकता है, there's a probability, right? हजारों जनों को कुछ नहीं हुआ, तुमको सीवियर इफेक्ट हो सकता है. There is a small minuscule possibility that you will get a very severe side effect. There is a possibility. It's not zero. If it happens to you, that's the chance you have to take, right? So that is where the naysayers are there. The benefit surely outweighs the vaccine. And if you read my book, there is a chapter on rotavirus. If you read my book, that addresses this issue of why people should take vaccines. How do you calculate risk-benefit ratio for a vaccine? And I'll tell you the last thing मैं बताता हूँ. देवराज, आप अब थोड़ा बहुत ये भी आपने सुना होगा, कोवैक्सीन और कोविशील्ड के बारे में सुना तो होगा, राइट?
Yes, sir.
आपने कौन सा लिया?
स्पुतनिक.
अच्छा, स्पुतनिक लिया. अच्छा, स्पुतनिक लिया या कोवैक्सीन? अगर आपको पूछे कि कोवैक्सीन अच्छा है या स्पुतनिक अच्छा है, आप क्या बताओगे?
I will be blank. I mean, because I have not experienced both.
But you've not experienced both, right? Let's say you were knowledgeable. Let's say you were an immunologist, hypothetically. Hypothetically, if you had studied immunology and somebody asked you this question, should I take Covaxin or should I take Sputnik? आप क्या बताते? मैं रिसर्च करके आता हूँ, मैं बताऊंगा ये अच्छा है कि नहीं है.
Data, something like that.
डेटा, हाँ. एक का 95% एफिकेसी है, एक का 92% एफिकेसी है. तो आप कौन सा बताओगे? स्पुतनिक का 92 है और कोवैक्सीन का 95 है. अब कौन सा, अब which vaccine will you tell?
95 going by the numbers.
हाँ, नहीं, फिर जो जिसका 95% एफिकेसी है ना, उसका 20% सेफ्टी साइड इफेक्ट्स है. ठीक है, और जो 92% वाला है ना, उसका 2% साइड इफेक्ट्स है. अब कौन सा लोगे?
This becomes a multi-dimensional problem.
Exactly, it's a multi-dimensional answer. There's no one answer कि ये वैक्सीन अच्छा है कि नहीं है. तो मैं क्या करता हूँ, मैं क्या बताता हूँ लोगों को कि कौन सा वैक्सीन अच्छा है? सब बेकार है. बाद में ही पता चलेगा यार, 1-2 साल के बाद ही पता चलेगा कौन सा अच्छा है. अब भी कोई ले लो, सब बेकार वैक्सीन है, बाद में ही पता चलेगा. ऐसे ही बताता हूँ मैं लोगों को. लेकिन कोई एक तो ले लो. We don't have enough data to say whether one vaccine is better than the other, है ना? And it's multifactorial, so it's very hard to say it's answers like this. Okay, any other question that's come there? Really good questions.
Yes.
Thank you.
Yes, and this can set all of us thinking. How was the COVID-19 vaccine developed so quickly? There are a lots of diseases which still existed before this virus. So how was this vaccine developed so quickly?
Well, since we have only 7 minutes more on this call and I have to go and jump onto another call after this, I'm just going to tell you a shortest answer I can give you. Read the COVID chapter in my book. I explain precisely how this vaccine was developed in 1 year.
Okay, thank you, sir. That's good. So,
But maybe since the call is asked, I'll tell you a few examples. The sequence of the virus was sequenced in beginning of February. Beginning of February, the virus was the virus was sequenced, which normally takes a long time to sequence a virus. सालों साल लग जाते हैं पूरा sequence मिलने के लिए, right? Because of the technology, एक हफ्ते में sequence हो गया, right? Sequence होने के बाद to decide how to make the vaccine from that sequence, 2-3-4 साल निकल जाते हैं. 4 दिन में vaccine बन गया. Because collaboration. Chinese scientists did the sequencing and American scientists made the vaccine. ऐसे collaboration कौन करता है? कोई नहीं करता. लेकिन कोविड में हो गया. 4 साल ऐसे ही बच गए. 2 दिन में 4 दिन में हो गया. जिसको 4 साल लगते थे. So like this, I can keep adding. Enormous amount of collaboration happened. That is why it was, and this is not normal, but really beautifully done. And that is why it was developed in a year.
That's amazing. That's a great point that we all of us should realize this: the contribution, collaboration, and communication of science, how important it is. And I think sir has just underlined the whole point of all of us being here today. Thank you very much, sir. This is amazing. I mean, this is a nice thing which you said, and I think we can't emphasize more why we are doing this activity.
Absolutely, Devraj. Absolutely, Devraj. Exactly.
So, sir, thank you very much for your time, and thank you very much for this session. It's been very exciting. And students also, thank you for interacting. Teachers also, thank you for the great interaction. And it has led us to a very nice conclusion. I'm very happy about the last remarks which sir has made. I can't express myself, but that's really great. Thank you so much.
You're so welcome. And thank you again, everyone, for coming. This is my alma mater, and this is where I started my journey. It's such a pleasure to be able to do this presentation in Junjunwala College.
Thank you. Thank you very much, Dr. Naren. Thanks a lot.
Yeah, thank you. Thank you. I'll come next time again in April to Junjunwala.
Sure. I wish the same session could have been face-to-face, the fun we had on 23rd.
We'll do that again. That time Janvi will be there.
Yes. Kuch to le lo. That was a good one.
Thank you,
Thank
sir.
you.
Thank you.
Thank you so much. Thank you, Kiran. Thank you for organizing this.
Yeah.
Okay. Bye-bye.
Thank you, sir.
Okay, so we'll now disperse. We'll break into our 4 sessions. Physics and chemistry will remain here, and others will join their respective sessions. We are planning to have some. We can take a screenshot. Yeah. I mean, we should have taken when he was here.
I have taken some with sir, but we can add the one with sir to what we take now and be a part of our photograph records.
Okay.
Yeah. So is it possible to make a view? I take it.
We were planning to have a couple of presentations here, but since this extended, we'll now directly go to parallel sessions. We'll have all presentations in parallel sessions.
Okay, so I think Kiran sir will guide us on how we proceed to next part of the program. Sir, please.
Yeah, physics and chemistry presentations will happen here in the same Zoom meeting. and other in charges, please start your Zoom session. And there are 4 parallel sessions. One is for physics, chemistry, other is for biotechnology, then there is botany and zoology, and there is humanities, right? So please join your respective session. Yes, Dr. Dubey is here, and Dr. Nita Srivastava, is she there?
I'm too here.
Yeah, okay.
Welcome, madam. Welcome, Professor. Thank you for joining us. Thank you for accepting our invitation to the program.
Thank you, Devraj. That was a very nice talk by Dr. Chirmule.
Yes, exciting. Adding
Much
new
very
knowledge to
interesting and
things
very
we did every
relevant.
day.
Hello, Dubey sir.
Namaste, madam.
Yes, sir.
Start, sir.
Yes, we can start. Who's the first? You can
Yeah,
make that student co-host, and
yes, sir. Just a minute. The first presentation for today is virus recognition and the proof of existence of genomic RNA.
It is in botany session, na?
Sorry. Yeah, so today's first physics chemistry talk is about quantizing eigen problem Schrödinger. Physics from physics.
In our judging sheet, Palak madam, we'll have the student's name. We can check his present absent.
Okay.
perhaps that is Jaswant Pandey, and he's not there.
Next session is on the law of the energy distribution in the normal spectrum.
Yes, sir. Sir, can you make me host?
Yeah, sure.
Hello, it is echoing little bit. I think it is echoing.
Now, Abhi, say you.
Yeah, it's okay, Gopi. You can proceed.
Yeah, student, can you just switch on your audio?
Yes, ma'am.
Who is presenter?
Sir, I'm not host. So can you make me host so that I can make student co-host? Devraj sir? Devraj sir?
I think Devraj sir is.
Hello. I
Devra
was
sir,
just.
can just make me host so that I can make host
Oh,
to the
yes.
student. Yeah.
Oh, sir. So, Palak madam, you are the host now.
Yeah.
Thank you very much.
Thank you, sir. Yeah. Gopi, na? Gopi Vishwakarma.
Yes, ma'am.
Okay, just a minute. I'll make you. Still, I'm not able to make you host. Just a minute. Huh? Gopi. So you are host now or co-host now. You can share your screen.
Ma'am, I'm starting now. I'm Gopi Vishwakarma from SYBSC. My topic name is on the law of energy distribution in the normal spectrum. ये जो पेपर है ये पेपर बहुत इंपॉर्टेंट था उस टाइम पे और इसने पूरी तरीके से फिजिक्स को चेंज करके रख दिया था उस टाइम पे इन्होंने ये पेपर इंट्रोड्यूस किया है मैक्स प्लैंक के द्वारा ये है लॉ ऑफ एनर्जी डिस्ट्रीब्यूशन ऑन द नॉर्मल स्पेक्ट्रम सो नाउ वी आर स्टार्टिंग फ्रॉम हियर उन्होंने मैक्स प्लैंक ने क्या किया था कि जो हमारा जो ब्लैक बॉडी का जो कर्व आता है एनर्जी डेंसिटी का इन्होंने एंट्रोपी के थ्रू कैलकुलेट किया था क्योंकि वेन स्पेशल लॉ और जो दूसरे लॉ थे उसको नहीं डिफाइन कर पा रहे थे तो उन्होंने एंट्रोपी के थ्रू इसको निकाला था एनर्जी डेंसिटी को तो एनर्जी डेंसिटी को निकालने के लिए उन्होंने पहले एंट्रोपी का उन्होंने फॉर्मूला दिया कि एंट्रोपी क्या होता है चेंज इन एनर्जी अपॉन से एंट्रोपी का चेंज एंट्रोपी अपॉन चेंज एनर्जी क्या होता है वो अपना 1 अपॉन टेम्परेचर के इक्वल में आता है तो पहले हम स्टार्ट करेंगे ना एंट्रोपी को कैलकुलेशन से तो उन्होंने एंट्रोपी के डेफिनेशन दिया था लेकिन वो डेफिनेशन अलाउनेबल नहीं था इसको कैलकुलेट करने के लिए एंट्रोपी को तो उन्होंने क्या किया कि जो एक पॉसिबल देखा कि जो अपना जो टोटल एंट्रोपी होगा वो हमारे टोटल एनर्जी के इक्वल में आ जाएगा ना मतलब टोटल एनर्जी पे डिपेंड करेगा और किसी रेजोनेटर के अकेले की एनर्जी पे डिपेंड नहीं करेगा तो उन्होंने इस तरीके से टोटल एंट्रोपी था वो इक्वल्स टू एन नंबर ऑफ द रेजोनेटर मल्टीप्लाई बाय एंट्रोपी ऑफ वन रेजोनेटर निकाला था उसी तरीके से टोटल एनर्जी जो कि एन रेजोनेटर इनटू मल्टीप्लाई बाय एवरेज एनर्जी ऑफ वन रेजोनेटर निकाला ना उसी तरीके से उन्होंने अभी क्या किया उन्होंने जो एनर्जी थी हमारी उसे क्वांटाइज फॉर्म करने की कोशिश की थी उसे क्वांटाइज करने के लिए उन्होंने एक एनर्जी एलिमेंट लिया और मल्टीप्लाई बाय पी जो कि एक लार्ज नंबर ऑफ इंटीजर लिया था तो इससे उन्होंने इसे क्वांटाइज कर दिया ना हम लोगों ने क्या किया कि जो यहाँ पे रेजोनेटर्स है यहाँ पर एक एग्जांपल है कि यहाँ पे 10 रेजोनेटर्स है उसके ऊपर उन्होंने एनर्जी को डिस्ट्रीब्यूट कर दिया है इस तरीके से ना अगर हम लोग एनर्जी को यहाँ पे डिस्ट्रीब्यूट करते हैं तो हमें कुछ इस तरीके से पिक्चर मिलती है ना इस पिक्चर को उन्होंने एक कॉम्प्रेशन बोला ना इस तरीके से कई सारे हमारे पास डिस्ट्रीब्यूशन हो सकते हैं एनर्जी के ना इस तरीके से कॉम्प्रेशन बोलते हैं ऐसे कई सारे कॉम्प्रेशन उनसे भी पॉसिबल कॉम्प्रेशन का अगर डिस्ट्रीब्यूशन निकाले तो हमें कुछ इस तरीके से हमें मिलता है जो यहाँ पे जो आ रहा है हमें टोटल नंबर ऑफ द पॉसिबल कॉम्प्रेशन बताता है ना इसके ऊपर अगर हम स्टार्लिंग फॉर्मूला अगर यूज करते हैं तो हमें कुछ इस तरीके से हमें जो टोटल नंबर ऑफ दी कॉम्प्रेशन मिलते हैं वो कुछ इस तरीके से मिलते हैं ना उन्होंने एक हाइपोथेसिस रखा कि जो हमारा टोटल एंट्रोपी होगा वो के मल्टीप्लाई बाय लॉग ऑफ ऑल द पॉसिबल कॉम्प्रेशन के इक्वल में आ जाएगा तो अगर हम लोग उसमें जो पॉसिबल कॉम्प्रेशन को हम लोग यहाँ पे सब्स्टिट्यूट करते हैं तो हमें कुछ इस तरीके से इस फॉर्मूले की पिक्चर मिलती है जो टोटल एंट्रोपी आती है अगर हम लोग इसे फर्दर सिंप्लीफाई करते हैं तो हमें टोटल एंट्रोपी आ जाएगी वो कुछ इस तरीके से मिल जाएगी ये हमारा जो था एन रेजोनेटर्स के लिए एंट्रोपी था अगर हम लोग एक रेजोनेटर के लिए एंट्रोपी निकाले तो हम लोग को कुछ इस तरीके से मिल जाएगा ना अब हम लोग क्या करने वाले, जो हमारा जो हमारा वेस्ट लॉ है उसका यूज करें आता है जो आएगा। तो अगर हम चेंज करें लैम्डा को न्यू से, तो कुछ इस तरीके से का प्रॉब्लम होता है। यहाँ पे इस तरीके के कोई को सब से स्टॉक कर रहे हैं, कुछ सब का हमारे एनर्जी होता है तो 1/30 के प्रोपोर्शनल होती है। Sorry, ma'am.
He is disconnected.
So, ma'am, visible है? तो हमें यहाँ पे किस प्रोसेस लॉ का इस्तेमाल किए थे ना। अगर हम लोग एनर्जी डेंसिटी को इस फॉर्म में भी लिख सकते हैं, अगर हम इसे थोड़ा सिंप्लीफाई करें तो हमें एनर्जी डेंसिटी का हमें नीचे वाला फॉर्म में भी मिल सकता है, एनर्जी डेंसिटी फॉर्म में ना। अब देखिए हमें यहाँ पे तो उन्होंने क्या किया कि जो इंटेंसिटी एनर्जी के बीच में उन्होंने अपने प्रीवियस पेपर्स में एक रिलेशनशिप निकाला था, फॉर्मूला निकाला था जो कि कुछ इस तरीके से आता है। और हमें पता है कि एनर्जी डेंसिटी और इंटेंसिटी के बीच में इस तरीके की रिलेशनशिप आती है ना। अगर हम उनके जो प्रीवियस जो पेपर की जो इंटेंसिटी है जो फॉर्मूला अगर सब्स्टिट्यूट करते थे एनर्जी डेंसिटी और एनर्जी के बीच में हमें इस तरीके से हमें एक रिलेशन मालूम पड़ जाता है। अगर हम लोग इस रिलेशन को हम लोग 7th इक्वेशन में सब्स्टिट्यूट करते हैं तो हमें कुछ एनर्जी का एज ए फंक्शन ऑफ टेम्परेचर और न्यू के बीच में मिल जाता है फॉर्मूला ना। अब यहाँ अगर हम लोग देखें तो यहाँ पे एनर्जी क्या है जो इंडिपेंडेंट ऑफ सी है, तो हम लोग इसे टेम्परेचर के फॉर्म में कुछ इस तरीके से लिख सकते हैं ना। अगर हम लोग अब यहाँ पे हम लोग क्या करना है, एंट्रोपी निकालने की कोशिश करेंगे, ठीक है। तो एंट्रोपी निकालने के लिए उन्होंने क्या किया कि जो एंट्रोपी का चेंज इन एंट्रोपी चेंज इन एनर्जी है वो 1/T के इक्वल में आता है। तो हमें अगर हम लोग यहाँ पे जो टेम्परेचर का जो हमें निकाला है फंक्शन अगर उसे सब्स्टिट्यूट करते हैं तो हमें एंट्रोपी मिल जाएगी इंटीग्रेशन के बाद, कुछ इस तरीके फंक्शन के फॉर्म में मिल जाएगी एनर्जी और न्यू के फॉर्म में ना। अब उन्होंने क्या किया कि जो हमें जो यहाँ पे एंट्रोपी मिला है वो हमें बताता है कि जो हमारा जो एंट्रोपी है वो डिपेंड करेगा हमारी एनर्जी और फ्रीक्वेंसी के ऊपर डिपेंड करता है ना। अब उन्होंने क्या किया जो हमारा जो था वो है एक एनर्जी, तो उसको हमने क्या किया कि एच और न्यू के फॉर्म में लिखा हुआ है। तो एच और न्यू के फॉर्म में लिखने के बाद हम जो हमारे पास जो एंट्रोपी का जो पहले प्रीवियस फॉर्मूला था जो एक एंट्रोपी का था रिजल्ट के, तो अगर हम उसमें सब्स्टिट्यूट करते हैं तो हमें कुछ इस तरीके से वो एक की एंट्रोपी मिलती है ना। अगर हम लोग इस ये जो फॉर्मूला हमें मिला हुआ है एंट्रोपी का, अगर हम लोग उसे इधर सब्स्टिट्यूट करते हैं 9th इक्वेशन में और उसे डिफरेंशिएट करते हैं पार्शियली विद रिस्पेक्ट टू एनर्जी, देन हमें कुछ इस तरीके से टेम्परेचर के फॉर्म में इससे मिल जाएगा। अगर हमने सिंप्लीफाई करते हैं इससे आगे तो हमें एनर्जी मिल जाएगी जो कि इस तरीके से आती है ना। अगर हम लोग देखें तो जो हमें एनर्जी मिली हुई है अगर हम लोग उसे सब्स्टिट्यूट करते हैं फॉर्म इक्वेशन 8 में तो हमें एनर्जी डेंसिटी मिल जाएगी। और ना अगर हम लोग इस एनर्जी डेंसिटी में हम लोग न्यू को चेंज कर लैम्डा को हटाकर न्यू डाल देते हैं, यहाँ पे न्यू को हटाकर लैम्डा डाल देते हैं तो हमें कुछ तरीके से मिल जाएगी एनर्जी का फॉर्मूला ना। अब हमें यहाँ पे क्या बचा हुआ है कि एनर्जी डेंसिटी हमें मिल चुकी है, तो हमें अगर देखने का है कि अब ये सही है या गलत, तो उसके लिए उन्होंने क्या किया कि जो एक एक्सपेरिमेंट उन्होंने कुलवंकर जी ने किया था तो उसका उन्होंने डेटा यूज किया गया है। तो इस डेटा में उन्होंने क्या किया कुलवंकर जी ने कि जो हमारा एनर्जी था जो टेम्परेचर 100 केल्विन पे उन्होंने निकाला हुआ है और उन्होंने उसके करस्पॉन्डिंग एनर्जी डेंसिटी भी कैलकुलेट करके दी थी ना। तो अब हम क्या कर रहे हैं जो हमारे पास एनर्जी डेंसिटी का फॉर्मूला है उसको यूज करेंगे हम लोग और उसे सिंप्लीफाई करने पे हमें कुछ इस तरीके एनर्जी डेंसिटी का मिलता है कुछ यहाँ पे वैल्यूज। अब ये भी हमारे पास एनर्जी डेंसिटी है और हमारे पास ये भी एनर्जी डेंसिटी है। अगर हम दोनों इन दोनों को इक्वेट करते हैं तो हमें एच और के रेशो में यहाँ पे मिल जाता है। के का रेशो में से अगर हम लोग वेस्ट डिस्ट्रीब्यूशन लॉ का भी यूज करते हैं अभी यहाँ है तो हमें कुछ इस तरीके से एक्सप्रेशन मिलते हैं। अगर इन एक्सप्रेशन को हम लोग फर्दर सिंप्लीफाई करते हैं तो हमें कुछ इस तरीके से एच के का रेशो मिल जाएगा। और इन दोनों अगर यहाँ पे भी हमें एच के का रेशो मिला है और इसका भी रेशो मिला हुआ है तो अगर हम इन दोनों को आपस में इक्वेट करते हैं और सिंप्लीफाई करते हैं आगे तो हमें एच और के की वैल्यूज मिल जाती है। यहाँ पे एच की वैल्यू हमारे पास वही प्लैंक कांस्टेंट है जो कि इन्होंने इनके प्रीवियस पेपर्स में भी निकाला था। तो इससे हमें ये मालूम पड़ता है कि जो एंट्रोपी के थ्रू हम लोगों ने एनर्जी डेंसिटी डिफाइन की है वो सही है। सही है, थैंक यू सर।
Judges, you can ask questions.
Yeah, Dubey sir, are you going to ask or shall I ask? Yes, can I ask?
Yes, madam. Yes, sure.
Yeah. So I was just asking, sir, can you ask first question or shall I ask?
No, you ask the question first. It's
Okay.
physics paper.
Yeah. Okay. Okay. So Gopi, it seems you have done a lot of work on this particular paper, lots of mathematics. So first thing, you tell me that have you understood what was going on and what you are actually doing in this particular paper? Because lots of mathematics you have done, you have correlated entropy with energy and proceeded very well. Have you understood that part? So I'll just.
Yes, ma'am.
So basically you were working with black body spectrum, right?
Yes, ma'am.
Black body spectrum. Is it applicable for all types of body which is emitting radiations, or it is only for a black body? Because any body which is emitting radiation, what kind of energy distribution you observe? Or it is only for black body?
जी मैम, ये एप्लीकेबल है।
एप्लीकेबल रहेगी क्या वो, मतलब ब्लैक बॉडी के अलावा भी किसी बाकी के बॉडी में? बिकॉज ये जो एनर्जी डिस्ट्रीब्यूशन है, दिस इज अ यूनिवर्सल लॉ, ऑलमोस्ट इट शुड बी एप्लीकेबल टू ऑल. ऑफ कोर्स द coefficient of emission comes into existence and but the
yes मैम, ये एप्लीकेबल है क्योंकि हम लोगों ने इसे यहाँ पे एंट्रोपी के थ्रू निकाला हुआ है।
हाँ,
सो यू कैन यूज इट अदर दूसरे ऑब्जेक्ट्स पे भी आप यूज कर सकते हैं इसका एनर्जी डेंसिटी कैलकुलेट करने के लिए उसकी।
एक मिनट, आप अपने इसमें जाएंगे क्या, स्लाइड्स पे जाएंगे क्या? बिकॉज बीच बीच में आवाज काफी कट रही थी आपकी. आई जस्ट वांटेड टू आस्क समथिंग.
मैम द कोर्स बनाई। Yes, ma'am.
Yeah, you have to, this is last one. You have to show me all the slides starting from. So you have used which mathematical expression here? That's it.
Which mathematical expression?
Yeah. Planck's or Boltzmann.
Stephen Boltzmann लॉ का यूज किया गया मैम यहाँ पे एनर्जी डेंसिटी का.
So because these can be done by Boltzmann statistics also, and then your Planck's black body radiation also, both. So which one you have taken into consideration? Boltzmann.
Yes, ma'am.
Show me the previous slide.
Yeah.
Yeah. Okay. how did you get this entropy relation?
मैम अगर हम लोग यहाँ नीचे ln को जो कि log है हमारा उसको
हाँ।
यहाँ पे सॉल्व करते तो हमें ये एक्सप्रेशन मिलेगा। इसका काफी लंबा वो सॉल्यूशन आता है तो
तो
उससे करने
मतलब
वाले
तुमने you have taken it from that particular book or expression, you have just taken the result.
मैंने सॉल्व किया है मैम इस एक्सप्रेशन को यहाँ पे मैंने
अच्छा,
और अपने बुक में से
Okay. And what is R over here, which you have written?
all the possible number of the combinations जो हमें यहाँ पे पीछे आती है डिस्ट्रीब्यूशन है इसे हम लोगों ने कॉम्बिनेशन बोला है एक पिक्चर को यहाँ पे तो ऐसे बहुत सारे हो सकते हैं वहाँ पर डिस्ट्रीब्यूशन तो उन सभी का हम लोगों ने क्या किया कॉम्बिनेशन नाम दिया है उन सभी का डिस्ट्रीब्यूशन हमने निकाला है कुछ इस तरीके से यहाँ पे हम लोगों ने स्टार्लिंग ऑफर का यूज करके हमें ये मिला है।
Okay, so you have also used some numerical analysis, or you have used some more statistical method. Am I right?
Yes, ma'am.
Statistical method. You have used some. You are using Stirling approximation also. Have you studied about Stirling approximation?
मैम उतना नहीं मैंने बस थोड़ा सा मैंने पढ़ा था इसके बारे में।
Okay. So where do you apply Stirling approximation?
Sorry.
No, because you have used Stirling approximation over here. That means you are using large number of particles. You are using large number of oscillators, or because you are basically explaining everything mathematically. You have not laid the condition whether they are rigid rotators or they are oscillators or they are reflectors, what are they? Anyway, it seems very, actually it was very good, and everybody needs some higher mathematical knowledge to solve the whole thing. My questions are over. Yes, sir, आप पूछेंगे तो पूछिए.
It's okay, madam. No problem.
Okay, Gopi.
Okay, Gopi, please stay here because we have to fill the attendance form. Don't exit from the meeting. Okay. So next, yeah, stay here in the meeting. The next participant is Sachit Upadhyay from TY. And the topic of the presentation is on the stability of the motion of Saturn's ring. Sachit, are you there? Sachit?
Hello, I am audible. Am I audible?
Yes, yes. I'll make you co-host. Just a minute.
Yes, thank you.
Yeah. Now you are co-host. You can share your screen.
Okay. Is it visible?
Right now, no.
And now?
No, it is not visible. Not visible.
Okay.
It's blank. It's coming black. You can share it again.
Yes, actually, I think my desktop got hanged. Yes.
Now it is visible.
Now it is visible.
Just go to the slideshow. Yeah.
Is it visible now?
Yes, yes, please go ahead.
Sorry for that delay. So I am Sachit Upadhyay from TYBSC, and my topic is on the stability of the motion of Saturn's ring, which was proved by James Clerk Maxwell. So before getting into actual paper, I would like to throw some light on preface. So before this paper was published, we already knew that Saturn has rings, but for most of the part, it was mysterious, since we didn't know how those rings are held there and what are they made up of. But sometimes discovery of certain things come from strange paths, because interestingly, these questions were tackled and proved mathematically first rather than direct observation by none other famous physicist James Clerk Maxwell, who we know is very well known for his major contribution in electrodynamics, but his hands like mutant too were in many ideas. So in this paper, he not only found out answers to the mentioned question above, like what the ring, why, how they are held up there, what they are made up of, but he also tackled questions: will those rings be there forever, or they would get disturbed and hence get destroyed? So again, I would like to also mention the timeline preceding works which provide stage to Maxwell's work. So it was Galileo first in 1610 who noticed the ring, but he failed to identify the ring, and the mechanism of instruments was not that precise at that time. Then comes the Christopher Wren. With the help of his 1652 studies, he identified those rings, but couldn't provide explanation for its appearance. Then comes famous again astronomer Christian Huygens, which was the first to suggest that Saturn's rings are detached from planet itself by observing the telescope which was designed by himself. Then comes the Giovanni Cassini, after which the Cassini spacecraft is named after him only. He determined that Saturn's ring was composed of multiple smaller rings with gaps between them. In 1787, then comes the famous mathematician and also physicist Laplace, proved that uniform solid ring would be unstable and suggested that the rings were composed of large number of ringlets or called as annular disks. So what was Laplace's conclusion? Laplace considers a ring of Saturn as a solid form of which is investigated as if it was a liquid, because that is better treatment and provides good equilibrium condition. And it that finds that if breadth and thickness of that ring of Saturn are very small in comparison with the distance from the Saturn itself, then the section may be ellipse. And it also, in the formula of proportion of axis, also admits that it is ellipse because it is considerably flattened. He also posits that rings were more like annular disk of concentric circle, which was mentioned previously, which is also well supported by his own theory. He had given remark on observation of few scientists who noticed that there are some irregularities in the ring. He said that if the ring were rigid and uniform, the slightest disturbance would cause it to fall on the planet. So there was heavy problem with the solid rigid ring system of the Saturn, and it is mathematical, so we will see how it happens. And here enters Maxwell. So Maxwell's first comment on Laplace's observation is that if rings were solid and uniform, their motion would be unstable and they would be destroyed. But they are not destroyed and their motion is stable. So he used the logic here. Therefore, they are either not uniform or not solid. So this paper by Maxwell is divided into two parts. The first part considered is about on the motion of rigid body of any form of shape about a sphere. Here we actually get into the paper. The first was to find the condition under which the uniform motion of ring is possible. Then second problem of this part was to find equations of motion when the rings get slightly disturbed. The symbols of the disturbance of coordinates are attached to the symbol of coordinates of the second problem here. So new symbols are likely to change with time with respect to, and we get series of linear differential equations of second order. And the third problem in this part, to reduce the three simultaneous equation of motion to form into a single equation. And there comes fourth problem, to determine whether the motion of ring is stable or unstable by the means of relation of coefficient ABC, which we get from the three simultaneous equation in the third problem. Now, here get little paper gets twisted because problems 3 and 4 give key of Maxwell's general method, and problem 3, the symbols of operation and quantities are separated. And we get new quantity here, n being written for d by dt. So differential gets eliminated, and which amounts the same, assuming that variation of any element of the ring may be represented by epsilon raised to n t plus alpha. So equation are thus formed which have no differential coefficient. Then it is remarked that supposing the value of n found, it will consistently generally of obviously real and imaginary term, the n. So what are the conditions for n? If n turns out, if for expression for n, the imaginary in that imaginary term turns out to be 0, then the variations of element will increase with time, and it definitely without the change in sign indefinitely. If both have same values, that is real and imaginary have some values, the variation of elements will be periodical with constant increase in the values of coefficient. And if the real terms get 0, the variation of elements will be periodical but with constant coefficients. So last mentioned case is the most suitable case mentioned over here to provide stability. And the relation of between A, B, C from third problem are to be determined, which will ensure the values of n can be purely imaginary. Then comes the second last problem, problem number 5, which determines certain quantities required in application of the theory to a rigid ring of constant and variable section. Because rings are, there are
Time is up.
sections.
Yeah. Conclude the session. Yeah. Time is up. Conclude, please.
So there was part 2 also on the motion of rings and the parts which are not rigidly connected, which shows it consists of many satellite problems. As we know, rings of Saturn have many small icy bodies, and these were those three equations which showed. And these are the wave-like structures we get in the Saturn's ring. So
Hello, Sachit.
Yes,
I'll have to interrupt here. We will have to keep to time for various reasons.
okay.
So
I will
you have done a good presentation, good studies, and I think our respected judges will discuss the part of the talk which you have presented. So I request you to conclude.
Okay, sir. Sir, this was the
Thank you,
maximum.
Sachit.
Yeah, please, judges, you can ask the questions.
No, let's give them time to conclude. Let's give them 1 minute after the warning so that they can say what they want. Yeah.
Yeah.
Conclusion slides, Sachit, you can say what was your conclusion.
Yeah, you can present your conclusion.
Yes,
Then
sir.
the discussion can begin.
Maxwell's conclusion was that at last he concluded that the rings cannot be rigid body, and there is the only one way possible that equations could hold, that the rings are actually composed of indefinite number of unconnected particles revolving around the planet with a different relative velocity. And also the fluid theory, which was previously stated by Laplace, got false out because when we observe Saturn, the Saturn was observed through the rings, and there was no medium available to note in the refraction or any such things was available. So there was noted no presence in the presence of medium which can refract light. So fluid theory also falls out, and this is how Maxwell concluded the structure of links and its stability. Actually, this paper involves heavy mathematical solutions of calculus of variations, which I am not going to because it is 92 pages long. Yes, thank you, sir.
Okay, good.
Thank you, Sachit.
We'll give them 1 minute after warning so that they can.
Mhm. Yes, Dubey sir and Neeta madam can ask questions.
Yes, sir. Dubey, sir.
Yes, madam. Sachin, thank you very much. Can you give some idea about the composition of the ring?
Yes, sir. They were icy rocks, obviously very easily available like helium, methane. But that was not the concern of this paper. This concern was the paper that those rings present are not rigidly ring-like structure, because at that time the scope of instrumental mechanism was not that good to observe. So they thought they were rings. So there was various theory that ring could be fluid that can contain partial gases, partial liquids. But it was Maxwell who showed that it must be small, indefinite numbers of satellites. Then only structure and stability of such rings can be ensured.
Okay, that was the main object of the paper at that time. But nowadays, everything is available. Everything is almost known.
Yes, sir.
And on the basis of your topic, you should know that what is the chemical composition of the ring, what is the chemical composition of the main body of the Saturn.
Yes, sir.
Have
That
you some
only
idea?
helium and methane will be most of the icy rocks that they found.
Okay. And what is the dimension of the ring? Means length and width and diameter, all these
Yes,
things.
yes, yes, sir. It is almost like 1,77,000 kilometer wide.
Okay.
Considering the A, because there are many parts in this ring. There are 7 parts in the ring, starting from A, B, C, and D to internally towards Saturn, and F, E, and G outwardly. They were fainter, so they were discovered later.
What is the main reason of its stability?
Because those rocks, yes, sir. Yes, sir.
Why it is still stable in the form of ring, not having any distortion or such things are there?
Sir, these rocks behave in such way. There is this one.
Okay, what is the level of gravitational level at Saturn?
Sir, I have no. That is the part of data which I don't have now.
It increases or decreases in comparison to Earth?
Obviously increases, sir. Gravity of Saturn is higher than Earth.
Okay,
And
thank
it
you.
must be high, because in later part of paper, which I cannot include in this PPT presentation due to time constraint, there is certain given numbers provided which I don't remember, that it must be greater than the rings of the Saturn's mass should be greater than the mass of the rings to hold it.
Correct.
And that ratio was that I forgot.
Okay, Neeta madam. Yeah, Sachit.
Please ask the question.
Yes, yes. Sachit, that was actually the paper is very nice, and the way you have proceeded logically to prove that it is not a it is not a metallic ring, but it is made up of rocks, and it is a collection of a tiny or big particles which are moving. So that was actually very nice presentation, but I feel we could have we could have time to see completely. I had some questions in my mind, but sir has already asked about its stability, gravitational field, and of course the composition. So good one. Only thing is that you should be able to compare it with the results obtained long time back and what it is right now. So you can always compare it. Okay, Sachin.
Thank you. Also, I would like to thank Kiran sir, because he supported me at the last time. I was when I was about to get over it. Thank you, Kiran sir.
Sachit, you can stop the share.
Yes, yes. Actually, my PC is getting little hang. That's
No
why.
worries, no worries. The next presentation is from Ujjwal Verma, SY student, SYBSc student, evidence for the 2 pi decay of the K2 meson. Yeah, Ujjwal, are you there? Ujjwal, I can't see.
Ujjwal is logged in as Gopi because he's using his device. He will just
Okay, no problem.
make a thing. You can
Yeah, I will make
allow
Gopi as a
yes
co-host. Okay,
co-host.
fine. Yeah.
Yeah. Thank you.
Yes, Gopi is now co-host. So, Ujjwal, you can share your presentation through Gopi's device.
Hello, I am Ujjwal, and in the
We can't hear you properly.
hello,
Ujjwal.
I am Ujjwal, and in next 8 minutes, I'm gonna try to express about this experiment, which is considered to be quite important in the physics community. And this experiment was conducted by Val Fitch and James Cronin, and it is something about related to CP symmetry. And in particle physics, there is something known as charge parity time, CPT. Parity symmetry means that the physics works the same no matter what's the location in the universe. Charge symmetry means that the interaction between charges does not get affected when the charges are swapped. Time symmetry means the physics look the same in the forward direction of time or the backward direction of time. And in 1950s, it was considered that CPT was correct and holds every time. But in 1956, Li and Yang pointed out that parity symmetry was never tested for the weak interactions. Considering that weak interaction, involved symmetry, Professor Shensung Wu and put it in a strong magnetic field, aligning all the nucleus together and seeing if the beta decay occurs in both the sides equally or prefers some direction. And if it prefers some direction, then the parity symmetry will break. Because, for example, why will the parity symmetry break? Because if you throw a ball forward in forward direction and put a mirror aside, you will see that ball will also go in forward direction. But in this experiment, the ball actually gone backward direction. बॉल आगे गई रियल वर्ल्ड में और मिरर वर्ल्ड में बॉल पीछे गई और दिस इज जस्ट क्रेजी। तो जो पैरिटी सिमेट्री लग रहा था कि कंजर्व रहती है, वो एक्चुअली कंजर्व नहीं रहती। और इस एक्सपेरिमेंट के लिए प्रोफेसर शेनशंग वो को एक नोबेल प्राइज मिला उसी सेम ईयर एज कंपेयर टू आइंस्टाइन, जिसको 1930s के बाद मिला था उसके 1905 के डिस्कवरी। और एक्चुअली ये एक प्रॉब्लम था जिसको फिजिक्स सॉल्व करना चाहते थे, और उन्होंने सोचा कि अगर वो जो बॉल पीछे गई, पार्टिकल जो पीछे गया, अगर वो एंटी पार्टिकल है तो मैच कंजर्व हो जाएगा वापस। तो इस एक्सपेरिमेंट में एक्चुअली जो अभी मैंने सिलेक्ट किया है उसमें यही बता रहा है कि वो जो पीछे पार्टिकल गया वो एंटी पार्टिकल नहीं पार्टिकल। और in when you take up, down and strange quark and combine them in certain arrangement, you get lattice strange mesons. Like this upper part, like you take anti-up quark, strange quark, you get a K negative, kaon. In particle physics, heavier particles decay to less heavier particle, and this is a pattern which occurs almost every time. और केऑन भी सेम पैटर्न फॉलो कर रहे हैं और न्यूट्रल केऑन्स डिके होते हैं दो टाइप से। एक में वो 2 पायंस में डिके हो सकते हैं, एक में वो 3 पायंस में डिके हो सकते हैं। और जिसमें वो 2 पायंस में डिके हो रहे हैं उनके पास काइनेटिक एनर्जी ज्यादा है, और क्योंकि उनके पास काइनेटिक एनर्जी ज्यादा है वो फील्ड्स अपने रिस्पेक्टिव फील्ड से ज्यादा इंटरेक्ट कर रहे हैं और ज्यादा जल्दी डिके कर अपने से छोटे मास वाले पार्टिकल्स में डिके कर रहे हैं। और जो 2 पायंस में डिके हुआ केऑन है उसको हम के शॉर्ट बोल रहे हैं, और 3 पायंस में जो डिके हुआ है उसको के लॉन्ग, क्योंकि के शॉर्ट के लॉन्ग से 550 टाइम कम एग्जिस्ट करता है। और जेम्स क्रॉनिन और फ्लिच जिन्होंने एक्सपेरिमेंट किया है, yes, okay, James Conner and Flitch conducted an experiment in which they shine kaon beam in a long tube, और वो लॉन्ग ट्यूब इतना लॉन्ग था कि के शॉट जब तक डिटेक्टर के पास पहुंचता वो दूसरे, और के लॉन्ग एक्चुअली एग्जिस्ट कर रहा है तो वो डिटेक्टर डिटेक्ट कर रहा है। तो एक्चुअली इस एक्सपेरिमेंट में क्या हुआ कि डिटेक्टर ने के शॉर्ट को भी डिटेक्ट कर लिया। इसका मतलब के लॉन्ग कन्वर्ट हो रहा है के शॉर्ट में, और उनकी रिस्पेक्टिव आइगन वैल्यूज एक्चुअली डिफरेंट है। तो जब वो इस इन इक्वेशन इन सीपी आइगन स्टेट्स में हम वो वैल्यूज डालते हैं तो वो सीपी सिमेट्री एक्चुअली एक्चुअली कंबाइन अगर हम तो वो हमेशा ट्रू होती है, लेकिन एक ऐसा एक्सपेरिमेंट है जिसमें टाइम सिमेट्री भी एक्चुअली टूटी है। और देख हो रहे हैं एक रिस्क है कि सीपीटी भी वॉलेट होगी, और अगर सीपीटी वॉलेट हुई तो ऐसा मैंने सुना है कि स्पेशल रिलेटिविटी और क्वांटम फील्ड थ्योरी रॉन्ग हो सकती है। और आई बिलीव कि सीपीटी ब्रेकेबल है, because best prediction, the worst prediction in all of science comes from the same theory, and that's it. And this credit because a lot of graphics मैंने वेरिटिज्म से उठाया। Thanks.
Thank you, Jewel.
Okay.
Neeta madam and Dubey sir, you can ask questions now.
See, first of all, I would mention here that I really don't know how far this student has understood the whole thing. Ujjwal, na? Yeah.
Yes, ma'am.
So about all these particles, okay, how they work. So I think a part of it only he has presented, and I don't know up to what depth he has understood, because the voice was breaking, and the voice was breaking for me, actually, for me. And I have understood very little out of it. Let me tell you very frankly. So if Dubey sir is going to ask anything, or Kiran sir is going to ask anything, that would be okay from my side. Because the annihilation of ions and what he was explaining, it was not very clear to me. Though the charge particle parity, time parity, all these parities are okay, but still.
Okay.
So
So,
Dubey sir or
Yes,
Devraj sir
actually,
can always help.
he was not audible for most of the time.
Yes, sir.
Normally, we got the video there. Ujjwal, what is the main role of meson in the nucleus?
meson, nucleus.
Yes.
Actually, meson combination एक quark. Nucleus में
Again, you are not audible.
है जो
He is not audible most of the time.
Please come in the range.
Okay, okay, sir. अभी audible हूँ?
It's little better.
Okay, sir. So meson actually quark और anti-quark का pair है, और न्यूक्लियस में प्रोटॉन्स और न्यूट्रॉन्स है, और प्रोटॉन्स 2 अप क्वार्क है, एक डाउन क्वार्क है, और एक न्यूट्रॉन 2 डाउन क्वार्क है और एक अप क्वार्क. तो meson एक्चुअली न्यूक्लियस में नहीं मिलेगा, शायद मेरे को कंफर्म मिलेगा.
So when you are talking about meson or about
Then
any
where it
particle,
is?
and you don't know where it exists or how do you get it, and first of all, the presentation should be such that even a layman person can at least understand what do you want to say.
Okay.
So must be very good. You must know about all these particles, for leptons, mesons, but things were not very clear.
It has very active role in stability of the nucleus, meson particle. Have you some idea about that?
No, sir, not much idea.
Oh.
And what are quarks? Just like that, I'm asking. What are quarks? Spin up quark, spin down quark.
Quarks are just elementary particle in standard model, and different combination of quarks and different particles give you bigger particles, like meson, protons, neutrons.
That's right. Okay, from my side.
So I should finish?
Yeah. So ask Dubey sir.
Dubey sir, do you have more questions? You can ask.
Please invite the next participant.
Okay. So Joel, you can stop the share. Yeah. So next participant is Sneha Chauhan from SYBSC. She will be talking on spectral classification. Sneha?
Yes, ma'am.
Sneha, are you there?
Yes, ma'am.
Okay, yeah, I'll make you host. Just a minute. You are co-host now. You can share your screen.
Ma'am, I can't share the screen.
So you have two, you have logged in two different names, Satu. So I'll make one more. You have, so I will make that as a co-host. Now see, both of your account are made as host, co-host. So
Yes,
now, yeah.
Is my screen visible to you all?
No, now it is visible. Yeah.
Yes. Good afternoon, everyone. Myself, Sneha Chauhan from SYBC, and I will be presenting on topic spectral classification of stars. According to European Space Agency, there are approximately 1 trillion 10 raised to 24 stars in the universe. The number for sure it will increase as human get better technology. But is there any specific way to categorize the stars in the universe? A well-established stellar classification to say? The answer is yes. Yes, the Morgan-Keenan classification system, an extended version of Harvard and Yerkes classification. Today's topic is based on MK classification. So by studying the spectra of stars, we can not only determine the color, but also chemical composition of star, the density of that composition, the temperature and luminosity, etc. So how do we find the spectra of stars? If you are seeing the star through your telescope and you place a slit over a focal plane of the telescope and you just attach a prism or grating to your telescope, it will give you a spectrum of characteristic shape of that particular star. And different stellar spectra have different appearance. In 1819, Edward Pickering and Fleming sorted star by decreasing hydrogen absorption line strength, where A-type star has strongest hydrogen line, then B types, and then C, and so on. The problem was that the other line did not fit into this sequence. 11 years later, in 1901, Annie Jem Cannon noticed that stellar temperature was the principal distinguishing feature amongst different spectra. She reordered the A, B, C types of hydrogen absorption line strength by surface temperature. The order of the stellar type are O, P, A, F, G, K, M, where O type star is the hottest with surface temperature of about 50,000 Kelvin, and M type star is coolest, having surface temperature just 2,500 Kelvin. The range of the temperature for each spectral class and color of the stars, which varies with the surface temperature, are shown here. And he used basically Wien's law of black body to classify stars based on the temperature. Since a glowing hot emission source like star emit a continuous spectrum that depends on its temperature, therefore star can be approximated as black body. Further, the stellar classification divided into subclasses from 0 to 9. The higher the number means the cooler star. Let's say the A-type star. Let's say in A-type star, A0 type star have higher temperature than A9, and he applied this Harvard classification scheme to about 22,000 stars and published as Henry Draper catalog. In this, it is an optical spectrum. Here we have various kinds of spectrum, spectral type on the left-hand side, running from O to M with couple of other at the bottom. On x-axis, we have plotted wavelength and the catalog listing of respective type of star on right side. Since we have approximated star as a black body, then according to the Wien's law, we can say that the hotter star appears blue, having temperature between 10,000 to 50,000 Kelvin. Medium hot star appears yellowish, and here we can see the O-type star are significantly brighter in blue, and M-type star are significantly brighter in red. So likewise, we can find the temperature of stars, and we can also see the kind of gradual progression of dark lines, that is absorption line. And A-type star are very strong in hydrogen line. By using quantum mechanics, one can determine at which temperature, which absorption line will appear corresponding to the different element. Hence, it is also possible to find the chemical composition of star. Now, if we plot the same spectrum and take wavelength on x-axis and intensity as a function of wavelength on y-axis, we observe the graph like this. Here, the hotter spectra type have peak wavelength, means the wavelength at which the emission is maximum lies in ultraviolet. And for cooler star, the peak wavelength is start shifting towards red. The Balmer lines H alpha, H beta, H gamma are much more deeper in the case of A-type star. And also the helium-1, which is a neutral helium in type B. Similarly, you can see that different type of stars having different prominent absorption line. Just assigning an alphabet to each star according to its surface temperature isn't enough for stellar classification. There are main sequence stars that are still burning hydrogen into helium in their core, and then there are white dwarfs that have ended their lives. So we need another parameter to differentiate them. That parameter is luminosity. In astronomy, the luminosity is total energy output per second. The MK or Yerkes system is based on two sets of parameters, a refined version of Howard from O to M scale and a luminosity scale of grade 1 for supergiant, grade 2 for bright giant, grade 3 for normal giant, and grade 4 for subgiant. And similarly for 5 for main sequence stars. Our sun is a G25 star. Its surface temperature is about 5,900 Kelvin, which shows G-type star, and it is fusing hydrogen into helium in its core, hence the main sequence star, which is shown by Roman number 5. The MK system come into play while plotting almost all of the stars in the universe on just one diagram, the Hertzsprung-Russell diagram, or HR diagram for short. Hertzsprung and Russell took a random group of stars and plotted the surface temperature on x-axis and luminosity on y-axis. They thought that this plot should be more or less uniformly distributed, but they found that the most of the star lies along the diagonally, a few stars above and few stars below. This is because the stars spend most of their lifetime in main sequence star phase. Now, this is the list of some stars and their spectral types according to MK system. In first column, we have listed the star in different catalogs. In second column, we have represented the name of the star. Now, why they have given such name? There are trillions of stars in the universe, and it is not possible to give them individual name. Let's take an example of Gamma Orion, where Orion represents it is a star of Orion constellation, and Greek letter represents the luminosity. Since Gamma comes after Alpha and Beta, therefore Gamma Orion is the third brightest star of the Orion constellation, which is Bellatrix. Let's take a real life example and understand this nomenclature. This is a well-known constellation, Orion constellation, captured by me. In this image, we have Alpha Orion, we have Alpha Orion, Beta Orion, Gamma Orion, where Alpha Orion is the brightest star of the Orion constellation, which is Betelgeuse, and Beta Orion is a Rigel, and Gamma Orion is a Bellatrix, and so on. Now let's see the spectral classification of these Orion stars.
Sorry to interrupt, Sneha. You have only 1 minute left.
Yes, ma'am.
So you can conclude. Yeah.
Yes. These Greek letters to the right represent the luminosity of respective star, and third column shows the spectral type of the respective stars. The first brightest star of the Orion constellation is Betelgeuse, having spectral class of M21AB, where M represents the temperature and color of the star, which is red, and 1AB denotes that this is the intermediate side of luminous supergiant, and so on. Thank you.
Neeta madam and Dubey sir, you
Yeah,
can
yes,
ask
madam.
question. Yeah.
Yeah, hi Sneha.
Hello.
That was a very attractive presentation. Very nice, clean presentation. I would like to ask you that from where did you From where did you get the catalog, or you took the catalog and studied on your own, getting the intensity and all?
This I got this from the research paper which I have took for
for your presentation.
Yes.
So have you understood the whole thing? The way they are representing intensity and
we actually we have studied the spectral classification of star. We get to know how do we take the spectra of the star, and just by looking the spectra of the star, we can tell the physical property of that star, like what is the chemical composition of star, and how what is the temperature, what is the luminosity and mass, and so on.
so on these charts, can you even tell how much time that light is going to take to come from star to Earth?
Which star, ma'am?
Any star. If you are studying these spectra and as you said, the intensity, luminosity, all other parameters. So can you also find how much time light takes to reach from that particular star to Earth?
Yes, ma'am. Like the Betelgeuse is 642.5 light years away from us. So light takes 642.5 years to come to the Earth. So Betelgeuse is a red giant, and it has ended their life. So it is it might be possible that it is already ended their life, and we are not able to see it.
Yeah, that is correct, because light which we receive simply tells us the history that maybe it existed or still existing.
Yes,
isn't it? That was again a very nice presentation. But one more thing I would like to ask you. Can you find out the size of the star, or how do you decide that it has entered into a red giant phase, or it will not die as a supernova or black hole?
This factor depends upon the mass of star. If star have a particular mass which is given by Schwarzschild radius, if that particular star have the radius more than that, then it will be after supernova, it will form black hole. Otherwise, it will form white dwarf, and so on.
Okay. Thank you, Sneha. Good presentation.
Thank you,
sir.
Thank you, Sneha, for very nice presentation.
Thank you, sir.
I would like to know one question, not only question, but one curiosity.
Yes.
Have you some idea about the probable diameter of the universe, observable universe? What is the size, approximate size of the observable universe?
It is around 46 billion light years.
46 billion light years. Very good.
Yes.
And what about the light years? The unit of the measurement?
Light years.
Kilometer?
What?
Can you
one light year is how many kilometers?
It is approximately 9.4 into 10 raised to 15 meters.
Okay. If suppose we are getting very low luminosity,
yes,
how it is connected with the distance of the star?
Luminosity and distance. Apparent luminosity, apparent magnitude, or intrinsic luminosity.
Okay. Have you some idea about interstellar space?
Interstellar space. Yes. this space, interstellar space. Sorry, sir, I don't know the answer at this time. I
Okay.
need to think on it.
Okay. Thank you.
Thank you, sir.
Thank you, Sneha. You can stop the share. Yeah, the next presentation is Akansha Singh and Khan Muhammad Alam from TY. Topic of the presentation is development of a method of polypeptide synthesis. Akansha, are you there?
Yes, ma'am.
Okay. Okay, I'll make a host both of you, or you are presenting from one device?
Ma'am, we are presenting from one device only.
Okay.
From security, madam, you can give share permission to everybody. Then you won't have to do it again.
Okay, sir. Yes, Akansha. Now
Yes, ma'am.
you can share.
Good afternoon, everyone. Myself, Khan Muhammad Alam, and my friend Akansha Singh. We would like to present our work on polypeptide synthesis. And this is the research paper of our topic, which we have referred to make this presentation.
Now here I will be representing the research paper which relates to the development of a method of polypeptide synthesis by Robert Bruce Merrifield. This is the abstract of the polypeptide synthesis. It has been studied that there is stepwise addition of amino acids which leads to a peptide chain bound by a covalent bond, and the big advantage was that when the peptide synthesis was done on a solid support, the desired product was obtained in short time and the yield of the product was high. The best proof for the polypeptide synthesis on solid support was the synthesis of ribonucleases. In this synthesis, sequence of 124 amino acids are linked to form polypeptide chain. Previously, synthesis of polypeptide was done in solution phase, but the disadvantage of this was that the yield of the product was less and purification was difficult. So in 1963, Robert Bruce Merrifield, he developed this technique. He received Nobel Prize for this impressive success in the year 1984. And this is the example of ribonuclease, which will be explained with the help of mechanism. So this is the mechanism of polypeptide synthesis, which was given by Robert Bruce Merrifield. This is the polystyrene, which is polymer, chloromethylated polystyrene, and we take one amino acid, and the N terminal of this amino acid is protected by protecting group, that is carbobenzoxy. And now this protected amino acid is attached to the polymer. Now it is important that to make this N terminal free so that we can add more amino acid, the deprotection of N terminal is done by removing the carbobenzoxy group. And the deprotection is done with the help of HBr in presence of acetic acid. Now the carbobenzoxy group is removed, and finally we get our free polypeptide. Now we will add one more amino acid from the left hand side. The N terminal of this second amino acid is protected by carbobenzoxy group, and this protected amino acid is now attached to this free amino acid. And now we will repeat the same step. Deprotection of carbobenzoxy is done with the help of HBr in presence of acetic acid, and to detach this polymer, we will treat it in the presence of base. And finally, we get our polypeptide, that is protein. Here we can see that when we combine this polystyrene polymer with amino acid, we get an ester linkage, that is RC double bond OR. And when two amino acids are combined, we get amide linkage, that with the help of removal of H2O, that is C double bond NH. Now, here is some other examples which I have mentioned. It is based on the same mechanism which was given by Bruce Merrifield. So here we took a resin support, that is a solid polymer, and this is our 2 amino acid. The N terminal of this amino acid is protected by protecting group, which is shown in yellow color. Now, to make this N terminal free, deprotection is done. So this is our free amino acid. Now we will take one more amino acid, that is protected amino acid, and now we will attach this to this free amino acid as shown below. So we get finally our, this is the peptide linkage and this is our amino acid. So to make the N terminal free, same step we have to repeat, that is HBr in presence of acetic acid, and to remove this polymer we will treat the whole compound in the presence of base. Now the next step was isolation and purification. Due to solid support, purification was easier, and desired product, which was obtained during the reaction, was chromatographed on Dweck's 50 column. Crystallization of peptide for purification was difficult because of high molecular weight and stickiness. In this method, peptides are purified by dissolving away the impurities. Due to this, desired product is obtained in short time.
As peptides form proteins, and we know that proteins are the building blocks of our body. There are many applications of polypeptides. First one is that peptides are used in discovery of drugs. For example, relaxofene, and this is used for the treatment of osteoporosis. If there is optimum level of peptides in our body, so insulin level is maintained, because we know that insulin is a peptide hormone. Peptides are also used in treatment of many skin problems. As we know, in today's world, the cosmetics play a huge role. So treat wrinkles, to increase the elasticity of the skin, to look younger. Different peptide and collagen based creams are used. There are many peptide hormones in our body, and they are required for the normal functioning of our body, like vasopressin, which maintains water level in cells, and oxytocin, that is birth hormone, and many more. Peptides also play an important role in many more fields, as shown the flowchart. Thank you.
The main reason for this approach was that the solid phase synthesis was convenient to be filtered and washed off. Time required was less. So by this convenient route, higher molecular weight polypeptide was obtained successfully. Polystyrene can be used as cation exchanger resin, and amino acid with chloromethylated polystyrene forms the most stable covalent bond. Last but not the least, these are the references from where we have collected the data of our topic and represented our work in front of you. Thank you and happy National Science Day.
Neeta madam and Subes sir, you can ask questions.
Yes, sir. Will you ask?
एक मिनट मैडम.
तब तक सर मैं एक क्वेश्चन पूछ लेती हूँ.
हाँ मैडम पूछिए.
Yeah. So this is peptide formation. Yeah, Akanksha and Khan Alam.
Yes, ma'am.
Yes, ma'am.
Yeah. So have you made this peptide?
No, ma'am. We have not yet. Actually, it was discovered by Merrifield. We have just saw the Nobel laureate lecture, and with the help of that, we mentioned this presentation.
Okay, that is right. So because all these polymers, I think peptide is a kind of polymers. They require some certain pressure or some desired temperature Am I right?
Yes, ma'am.
To link one bond with another bond, and very nicely first that one bond is protected with the presence of HBr, and then the same thing you are removing with the help of base. So acid and base continuously you are using in the in this formation.
Yes, ma'am. Because if we want to increase the chain length of peptide, so if we will not deprotect the one terminal, so reaction will happen from that side also. So if we deprotect it, so we can get our desired product, whatever we are means whatever is needed at that time.
So how long you can make a chain? That means what would be the approximate length, desired length? Up to what length can we go for? How big a molecule we can make?
Ma'am, exactly we can't say, because according to our presentation during Merrifield, when he synthesized this technique, ribonuclease was the most best proof that in that 124 amino acids total, they were linked to form a long polypeptide chain. So that was the number of 124 amino acids, which was.
Yes, sir, please ask. These questions I have just asked out of my curiosity.
Nana, it was very good question. And I would like to congratulate Akanksha and Khan Alam for having very excellent presentation.
Yeah, that was very nice, and I could understand it. That's why I say we all can say it was a very good presentation. Being person from physics, still I understood the gist and the base of how they have done the entire process. Congratulations, Akanksha.
Thank you, ma'am.
But Akanksha,
yes, sir.
During presentation, whatever you presented, that was the work of Bruce Merrifield.
Yes, sir.
So you should say that it was taken by the Merrifield. He observed, he presented, he did, not we did.
Yes, sir.
Correct, na? And
yes,
the second part was application of the polypeptide. So can you have some idea about polypeptide? Can you define it? What is polypeptide?
Sir, polypeptide means it will have means more than one peptide bond. Because when we combine two amino acids, in that we get one peptide bond by the removal of H2O.
Okay,
so
what is that? What is that peptide bond?
Sir, C double bond O NH.
NH. Correct, na? Amino group from one amino acid, carboxylic from another, combined to form the peptide bond.
Basically, it is a amide linkage.
Okay, it is amide linkage.
Yes.
Particularly in, you were talking about in solid state, it was prepared.
Yes, sir.
What is was main function of the blocking groups, typing groups?
Sir, actually the main function of the protecting group was that for some time it deactivates that N terminal, because if we will not deactivate that, so reaction can happen at the same end. We can attach the polymer from N terminal also. So to stop that, we deactivate that N terminal with the help of protecting group.
Okay. In human body, is there also synthesis of polypeptides and proteins?
Yes, sir.
How it occurs? In which organ?
Sir, in means like adrenal glands, kidney and all, we have different types of proteins and hormones. They are the part of peptide bond only. For example, if we will not maintain proper insulin level, so we'll suffer from diabetes, and that insulin itself is a peptide hormone.
Okay.
So
suppose insulin is consisting of polypeptide, correct, na?
No, sir, it doesn't contain that polypeptide. Means it is observed that there is one part which is known as C peptide. If in human body that level is optimum, so insulin level is maintained. Otherwise, because of that, maybe a person suffer from diabetes.
Okay. I want to know where is the site for synthesis of polypeptide or protein in human body. What is the source of amino acids in our body? The precursor is amino acid. From amino acid, there will be synthesis of the polypeptide and protein. What is the source of amino acids?
Ribosomes, there is protein synthesis.
No, that is another answer. What is the source of amino acid in our body that acts as a precursor?
Sir, means in human body.
In human body, in our body.
So from that we get vegetables and meats.
Whatever food we take, if there is protein, that is the main source.
Yes, sir.
Correct. And where its synthesis occurs? He was telling something about some.
Yes, sir, in ribosomes.
Okay. Okay, thank you.
Okay, sir, thank you.
Thank you, Akansha. So the next presentation number 8, Mariam Chogle and Aina Singh. So this presentation is
Mariam is with next.
So this presentation is not now happening. So Mariam is shifted to the presentation number 10, Akash Mahapatra. So I'm going to the presentation number Raisa Kasu and Anmol Bhagat from TYBSc. The topic of presentation is use of isotope as tracers in chemical research. Anmol, are you there?
Yes, ma'am.
Yeah, so I have already made you host.
Ma'am, please make co-host to 625 Raisa Kasu.
Okay. Okay, now both of you are hosts. You can share screen. Yeah, screen is visible. You can go ahead.
Okay, ma'am.
Good evening, everyone. I am Raisa Kasu.
And I'm Anmol Bhagat.
We are here to represent the research paper on uses of radioactive isotopes as a tracer in chemical research. So this is the first page of the research paper. So let's start. To understand the concept of radioactive tracers, firstly, let us see about the isotopes. Isotopes are the elements having same atomic number but different atomic mass number. And they are classified into two categories, that is radioactive isotopes and non-radioactive isotopes. For radioactive tracers, let us understand about the radioactive isotopes. Radioactive tracers are also called as unstable isotopes as they contain unstable combination of proton and neutron so that they emit radiation in the form of alpha rays, beta rays, and gamma rays. Radioactive isotopes are further classified into natural radioactive isotopes and artificial radioactive isotopes. The examples of natural radioactive isotopes includes tritium, carbon-14, and lead-210, and that of artificial radioactive isotopes includes chromium-51, cobalt-60, and technetium-99N. Now, let us understand about the radioactive tracer. The radioactive tracer is a chemical compound in which one or more atoms have been replaced by a radioisotope. The decay of radioactive tracer can be used to explore the mechanisms of chemical reactions and to study about the metabolic pathways in plants as well as animals. The idea of radioactive tracer was given by Dr. George D. Halsey, the father of nuclear medicine. As Dr. George D. Halsey was the failure in separating radium-D from lead chloride, but this was the boon for his success as he used radium-D as a tracer, which is now known to be the radioactive isotope. He was the first to apply both natural and artificial isotopes as tracers in studies of plants and animals. For his work, he received the Nobel Prize in 1943 and the Atoms for Peace Award in 1959. Moving ahead, let us understand how the concept of radioactive tracer in chemistry was born, or we can say that how Dr. Halsey got this idea. There comes a story of two great scientists, Rutherford and Dr. Halsey. One day in cellar of Rutherford Institute, Rutherford said to Halsey, "My boy, if you are worth your salt, you will separate the radium-D from all the nuisance of lead. As radium-D is an isotope of lead, it will have same chemical properties as lead. But this thing was not known at that time, and Halsey accepted the challenge to separate mixture of radium-D from lead chloride. But after trying a large number of separation, Halsey failed entirely to separate mixture of radium-D from lead chloride. To make the best of this depressing situation, Halsey decided to make use of inseparability of radium-D from lead chloride. And from here, the concept of radioactive indicator chemistry was born. Over to you, Anmol.
Thank you, Raisa. Okay. Proceeding further with the method of tracing. There are methods, we have mentioned 4 of them. First one is precursor product sequence. Second is double and multiple labeling. Third, competitive feeding. And fourth, isotope incorporation. How the concept of radioactive tracer is used in a case of living organism and in chemical reaction. Firstly, we know that plant perform photosynthesis. In this reaction, oxygen is formed as a byproduct. To know that oxygen molecule contain oxygen atom either from carbon dioxide or from H2O. When water enriched by the oxygen isotopes is used in a photosynthesis, it is found that oxygen involved in a process is come entirely from water, while the oxygen of a carbon dioxide is retained in organic compound. And the second reaction, that is hydrolysis of ester, it is found that acid produced in a process comes from the water, while the oxygen of the ester is retained in the alcohol. Yeah, the application of a radioactive tracer. First, in medical field, that is medical tracer, a chemical tagged with a tiny amount of radioisotopes and injected into the body, and the process through the body can be detected using a sensitive gamma ray. So you can see these organs and processing chemically correctly. Okay, for the goiter treatment, we use the iodine 131. Okay, used as a thyroid. And the second, in the sterilization, the high doses of radiation can kill the bacteria. So the synergid bandage extra sealed in a package will remain the sterilized with the help of this radiation. And even used to sterilize the surgical instrument using cancer treatment, food irradiation, and radiography. Here are the some reference.
Thank you so much for the patient hearing.
Thank you, Anmol and Raisa. Nita madam and Dubey sir, you can ask questions.
Sir, ask. Please ask. Dubey sir? Okay, then shall I ask few questions here? Again, I have very nice presentation. Let me tell you, last presentation also was very nice. The way you people are presenting each and
Thank you,
every
ma'am.
term is clear to me, at least how we are, how you all are
Hello.
presenting. Yeah. So can you just move your slide ahead so that I remember I can ask you the question. Now,
Yes, ma'am.
yeah, just next. Again, next slide. Next slide, please. Yeah, the previous one. Okay, so in this photosynthesis and hydrolysis, the previous one, you have mentioned that the oxygen which is liberated in this reaction, carbon dioxide and H2O. So water is, oxygen is coming from water, and the oxygen which is coming out from carbon dioxide is becoming a part of glucose, right?
Yes, ma'am.
How did you come to know that?
Yeah, ma'am, we come to know this because of that we had used isotopes of oxygen. We has introduced oxygen isotopes in water molecule.
Achha, okay.
And we already know that isotopic.
Okay. Yeah. And that you are using as tracer.
Yes, ma'am.
Okay. And next, please, next. Because I think that is the same thing. Okay, so my next question is in medical field where you are applying it. Can you just show me that particular slide where you have shown, you have mentioned, just a second. Yeah, you have mentioned here, yeah, that chemical is tagged with tiny amount of isotope and injected in the body. Now, in the body, how it is guided, where it has to go, in which organ it has to go, because it is tracer, it will only tell you that the
Hello, sorry,
drug
ma'am.
is.
We can't hear.
I just want to ask you that once you have tagged it, how do you guide that chemical to the targeted organ?
Ma'am, we are not guiding that chemical element. It's doing its function only. Okay, we are only just taking or tracing it with the help of the machines. That GM counter.
You are only tracing it, am I right?
Yeah, ma'am.
But it must be moving towards the targeted organ. So is there any way to take it to that particular organ? Because there are many organs up to which
Okay, ma'am.
The drug delivery is really very difficult.
Okay. Ma'am, it's like that key as iodine.
Yeah.
Okay, just sake of example, when we eat something, okay, if it's iodine is present in that, then iodine, most of the iodine will enter in thyroid.
Correct.
Because thyroid gland has that property to take up iodines.
Okay.
Then the radioactive iodine will also be enter in that part, and we can trace that.
And how do you treat cancer cells with that?
Ma'am, can treat by that gamma rays. As a radioactive isotopes releasing the gamma rays, which is very harmful, that can kill the cancer cells. That gamma ray has that much energy or properties that can kill the cancer cells.
But it also kills the RBCs simultaneously.
Yes, sorry, ma'am.
It also kills the RBCs, red blood cells. So not only the cancer cells, but the good cells also it kills, because it cannot distinguish.
Yeah, ma'am, but it's okay. Na, ma'am, our main target is to kill that cancer cells. If some trace amount of good cells also will kill, it's okay. Because our main target is to remove that cancer cell, no?
Yeah, but it is not workable when you have second level or third level of cancers. Anyway,
Yeah.
that was a good presentation, tracers and all. I
Thank
have
you,
come
ma'am.
to know for the first time. Okay.
Thank you, ma'am.
Thank
Yeah. So please ask.
Thank you, Anmol and Raisha, for a very
Yeah.
good presentation. Can you tell me that when you are using iodine 131 as a tracer element to investigate the functioning of thyroid gland, in which form, chemical form, iodine will get assimilated? Which chemical is present there, containing iodine in your thyroid gland?
Yeah.
Iodine will enter into thyroid gland in the form of pure iodine or in the form of some chemical, some metabolic product.
So in a form of some metabolic product.
Which there is some hormone or some material containing iodine. Can you name the chemical? How we diagnose the thyroid disorder? There are some hormones where iodine is present in the form of its chemical constituent.
Thyroxine.
Okay. T3, T4, TSH.
Yes, sir. T3.TSH.
What are that? T3, T4, TSH.
Stimulating hormone.
So they are
T3.
hormone.
T3. What does it mean? T3 means what?
Triiodothyronine.
Triiodo. Okay. T4.
That trioxine.
Tetraiodo. And what about TSH?
Thyroid stimulating hormone.
Thyroid stimulating hormone. Correct. Thus, iodine will get incorporated in the form of these hormones, or it has another role to play there?
No, sir, only in this form.
Which form? In the hormones form, molecule form?
Hormones form.
Yes.
So in hormones form.
Okay. And when we use cobalt-60, which rays are emitted from cobalt-60 that is used for sterilization purpose?
Gamma rays.
Okay. We are exposing here some fruits. Means it is being exposed with the gamma rays.
Yes, sir.
Okay. Can you give another 2 examples of use of cobalt-60 in chemotherapy?
Sir, for sterilization, for sterilization of instrument.
Okay, very good.
And in radiotherapy.
Okay, in radiotherapy.
Yes, sir.
Means radiation therapy.
Yes, sir.
Okay. Okay, thank you.
Yes, thank you, sir.
Thank you, Anmol and Raisa. So next presentation is Akash Mahapatra and Mariam Chaugale. The judges, please just add Mariam Chaugale also in this presentation. Akash Mahapatra and Mariam Chaugale from TYBSc. Topic is research on carbohydrates and vitamin C. Yeah, Akash and Mariam, I already made you host. Yeah.
Am I audible?
Yes.
Yes, ma'am. Yes, sir. Good afternoon, respected judges and all my dear friends present here. I am Ms. Mariam Chogle.
I'm Akash Mahapatra.
I'm very happy to present a research paper on this National Science Day. So our topic for today's Anusandhan research paper presentation is structural elucidation of vitamin C, for which Sir Norman Haworth received a Nobel Prize in 1937. But today we are going to focus only on vitamin C. This is the first screenshot of the first page of our research paper. This is the abstract of a research paper. This paper explains how backward integration of vitamin C structure was done to find out the exact structure, because that time spectroscopy technique was not there. So they had to break the molecule, and from the degradation products obtained, they arrived at the correct structure. So this is what this paper represents.
He named this compound hexauronic acid initially because at the time he only knew that the carbon chain contains 6 carbon atom, and then he gave the molecular formula C6H8O6. So what are vitamins? Vitamins are the organic substance that must be supplied to permit appropriate growth in the living beings or the maintenance of the structure of. Vitamin C, better known as ascorbic acid, is water-soluble essential vitamin, a potent reducing and antioxidant agent. Ascorbic acid has anti-ascoric properties. Anti-ascoric means cure the scurvy. Here, scurvy means scurvy. Hence, the name ascorbic acid was given. Has L isomer is active. D-ascorbic. There are two isomers. L-ascorbic is active and D-ascorbic acid. The actual structure of ascorbic acid was not known. Hence, we are just writing the name ascorbic acid. Ascorbic acid on the reaction with neutral iodine in a water give oxidized ascorbic acid along with a byproduct hydroiodic acid. This oxidized ascorbic acid, when comes in contact with HI, again gets reduced to form ascorbic acid. Due to this reversible oxidation reaction gives ascorbic acid its antioxidant property. This oxidized ascorbic acid on further oxidative cleavage using sodium hypoiodide gives a 2-carbon compound oxalic acid and a 4-carbon compound trihydroxybutyric acid. This trihydroxybutyric acid on further oxidation gives tartaric acid. From this reaction, he came to know that this reaction 2 carbon plus 4 carbon show that the ascorbic acid is 6-carbon compound, and from this and this structure he came to know that the ascorbic acid has 1 primary alcohol and secondary alcohols.
At that time, it was thought that the oxidation was due to the introduction of 1 oxygen atom rather than of 2 hydrogen atoms, or removal of 2 hydrogen atoms. Therefore, the following structures were put forward. The keto form of
Your voice is not audible. Yeah, Mariam, we can't hear you.
Sorry, ma'am, we got disconnected from another device. At that time, it was thought that oxidation was due to the introduction of 1 oxygen atom rather than the removal of 2 hydrogen atoms. Therefore, the following structures were put forward. The keto form of ascorbic acid gives absorption at 265 to 260, and its acidic enol form gives absorption at 245. Hence, it was concluded that these structures give keto-enol tautomerism. Another alternative formula of hydrofuran, that is a cyclic ester, was proposed. However, it failed to account several chemical properties and crystallographic data with vitamin C. Malic acid on oxidation gives dihydroxymalic acid, whose absorption spectra was seen to show similarity with ascorbic acid. Hence, it can be said that similar structure having a double bond and 2 OH groups is also present in ascorbic acid.
To find the exact constitution of ascorbic acid, further investigation shows that the structure can be best expressed from formula 4, which can be interchanged into formula 5 via enolylation, which is a continuous step and cannot be controlled, which gives a vitamin C. Its unique antioxidant activity. Both these structures give the similar reaction. This formula was further supported by the fact the freshly prepared vitamin C exists as a lactone, the structure 6, that is a diketo form of rather than a free acid, and when keeping in for some time gets converted into enol form.
This structure 5, that is the enol form, on reaction with acetone forms its acetone derivative, which is a cyclic ketal formed at the open end. This structure 5, that is the enol form, when reacts with trityl chloride, forms its trityl derivative, as trityl chloride is a protecting group for alcohols and reacts selectively only with the primary alcohol, which means that primary alcohol is present at the 6th position where CH2OH is present. When this structure 5 is reacted with diazomethane, then methyl groups get added to the enol position to form the dimethyl derivative. These reactions gives the evidence of the presence of 2 and 3 enol linkage.
Confirmation of 1,4 linkage was done using ozone. He used tetramethyl derivative, and in presence of ozone, 2,3 enol linkage is cleaved with the addition of oxygen to give a diester. This diester, when further reacted with ammonia, gives oxamide threonamide. This oxamide is a 2-carbon compound and threonamide is a 4-carbon compound. So in this structure, the presence of a free hydroxy group at the alpha position confirms the presence of oxygen linkage 1,4 position, and this reaction confirms the presence of 2,3 linkage and 1,4 linkage oxygen.
Conclusion: synthesis of vitamin C, which helped the world prevent and cure scurvy. Vitamin C tablets were synthesized, which helps to boost the immune system. Earlier, scientists didn't have the access to modern structural elucidation technologies like NMR. During this research, the integration of the results from the degradation product analysis to the similarity in chemical properties to the selectivity of reagents towards the product of the reactions, all these details were used by Mr. Norman Haworth just to arrive at the correct structure of ascorbic acid. This gave us a complete overview of how these degradation products were earlier used by scientists to integrate the knowledge and to build up the molecule. These are the references. Thank you so much. We are very grateful to get this opportunity to present this research paper.
Thank you, Mariam and Akash. Neeta madam and Dubey sir, you can proceed with the questions.
Okay, madam, I'm asking one question only, that the way the structure of vitamin C was obtained by the reverse method. Is there any other complex structure is been deciphered rather? It's, very good thinking to move in the backward direction and get the structure of the whole.
Ma'am, sorry to understand your question. Means any other complex compound which was used by this time, vitamin A was also discovered. The structure of vitamin A was also discovered by Mr. Richard Cohen using this degradation analysis method.
Okay, that was my question.
Yes, ma'am. So vitamin A was also, the structure of vitamin A was also analyzed and put forward. The correct structure of vitamin A was put forward by Mr. Richard Cohen using this degradation method.
Very useful method. So today, sir.
Okay. Can you let me know the chemical formula of vitamin C?
C6H8O6.
Okay, it is acidic in nature or alkaline?
Acidic, sir.
Why? Why it is acidic?
So because of its antioxidant properties, if it's become acid.
No, it has some carboxylic group or any functional group, such functional group.
Yes, sir.
Okay.
Yes, sir. Carboxylic acid is present, which gives it the acidity.
Okay. You told me that because of some antioxidant property, how vitamin C, that is ascorbic acid, is capable to work as an antioxidant?
So because of enolization, a continuous reduction and oxidation takes place between the between the diketo form and the enol form of ascorbic acid, which gives it its antioxidant property.
Okay, suppose there is free radical in our body, oxide, superoxide ion, O2 minus. How vitamin C is capable to neutralize it? O2 minus superoxide ion plus vitamin C, C6H12O6. How it is capable to neutralize the superoxide ion? It will work there as antioxidant or some other role. Okay, what is the modern technique to elucidate the structure of vitamin C?
So NMR. Using NMR spectra,
Okay.
vitamin C structure can be elucidated.
During this COVID period, every doctor used to recommend vitamin C. What was the reason?
Because its property to boost its immune system of the body.
Okay. The same is related with superoxide neutralization. How it boosts the immune systems. Okay, thank you, Akash and Mariam.
Thank you, sir.
Thank you, sir.
Yeah. So the next presentation, Shubham Jadhav from MSc Part 1, the discovery of Grignard reagent. Shubham, are you there?
Actually, sorry, ma'am. Hello.
Yes, yes.
मैम एक्चुअली मेरा प्रेजेंटेशन था अभी.
Then
Does the inertia of a body dependent upon its energy content?
Yes.
So
in the key, it is the inertia of a body depend upon its energy content. That's the given in the list.
okay. It's given. Yeah. Ansari Muhammad Adnan. Yeah, sorry. I just
Okay, no
missed it. Yeah.
problem, ma'am.
Yeah. So the Ansari Muhammad Adnan from TYBSc. Does the inertia of a body depend upon its energy? Yeah, I'll just make it. So you have joined from Adnan, na?
Yes, ma'am.
Yes, you can share your screen now.
Thank you, ma'am. So, hello everyone. My name is Ansari Muhammad Adnan, and my paper presentation topic is: does the inertia of a body depend upon its energy content? It is basically E is equal to mc square, Newton's famous equation. So I would like to start my presentation with a very good and simple example, like if we come like in hydrogen atom, if we weight the hydrogen atom, this mass, and if we broke the hydrogen atom and mass the proton and electron differently, and then we combine, then we found the hydrogen atom in which we didn't differentiate the proton and electron mass is less than the if we differentiate hydrogen atom in proton electron is less than. So it is how can it possible? So it is possible by the Newton's law is equal to mc square. So basically the point because of it is sorry, I'm sorry. Yeah, so we are going to look for the most famous equation is equal to mc square. So its mean energy, Newton says that energy is equal to a mass time of the speed light. This equation says the energy and mass matter in interchangeable. We can convert mass into energy and energy into mass, and in different forms also. So is equal to mc square in Newton's research paper, it written as m is equal to c by m is equal to e by c square. It is because that Newton's wants to focus what is mass actually is. So according to the Newton's according to Einstein, the mass is actually mass is not which mass we measure is not actually exactly mass which we measure, because there are or any other things which we have to measure for calculate the correct marks. Just like if there are two blocks of the same atoms, if we measure them both, but they can't be equal in the mass, because mass is depend upon how particles are arranged and how those parts moves within the bigger objects. For example, I have this for example a 1 minute, ma'am, it is not sliding properly. For example, we can take two flashlights which are similar and they are fully charged, and when we start one torchlight and it start emitting light, when it start emitting light, its electrochemical energy start to get removed by the light. It's converted into light and removed by the torch. Then if we measure this both of the mass, it will the light which is the flashlight which is emitting light is less less in mass, and the which is fully charged but not working, that torchlight has large mass. Reason because according to the Einstein's is equal to mc square, there are some extra component in mass which we have to measure, because as we show energy and mass are interchangeable, and they are in energy and mass are interchangeable, and we can convert into other. So according to Einstein, the energy is also we can think it as a mass. So so as we see, mass extra is equal to every type of energy C2. So as we see in torch example of flashlight, the electromagnetic energy is getting converted into light and it's getting out from the torch. So that's why its energy is decreasing, and energy is decreasing. That's why mass is decreasing also. And because C2 is very huge in number, it is very less, you can say millionth of percentage of total mass. So it is like we can't measure in by open eyes, we have to measure it in proper scale for proper setup. So that's why flash A is very less in mass with flash B. If we neglect the extra mass, then we can say it's equal. And the same example goes for the electron, which we talk about in start about for atom of hydrogen, in which if we break the atom and we weight hydrogen, we weight in electron and proton differently, then we combine the weight mass of both the particle, then we find the masses, they are different, and the revolving when electron is revolving around the proton, that mass is different, because the when electron is revolving around the proton, it has some kinetic energy, and because of the attractive when proton attract the electron, it converts sorry it when we then sorry then when electron is revolving, it has kinetic energy, and also it has running potential, or it, and also it has some type of potential energy. When two particles attract each other, it has some potential energy, and potential energy is negative in this case, and kinetic energy is not that much that it cannot change the potential energy negative effect, so that we calculate the total mass when electron is revolving around the proton, it gets the mass is less than if we measure it in different other different particles. Same goes for all periodic tables in our chemistry. And thank you.
Thank you, Adnan.
Thank
Neeta
you, ma'am.
madam and Dubey sir, you can ask.
Dubey sir, please ask.
E is equal to mc square. It was given by Newton or by Einstein?
Einstein, sir, sorry, by mistake.
Okay. E stands for energy.
Yes, sir.
And C for?
C for light speed, sir.
Light means what?
Sir, speed of light.
Speed of light. What is its value?
3 into 10 raised to 8 per second.
8 unit क्या है? What is the unit?
Meter per second.
Meter per second.
Yes, sir.
Okay. Can we convert energy into matter also?
Sir, energy into matter, be like when we do like we know that there are nuclear bombs in which we convert mass into energy. So it is known that we can convert the mass into energy in a very high amount of energy, but I don't see any experiment in which they have converted energy into mass till now.
Is it possible according to this equation or not?
Sir, if this question says mass and energy are interchangeable, then it's possible.
Okay. You said that mass of hydrogen atom is less than the mass of the neutron and proton and the electron. Why it happens?
Sir, because neutron, proton, electron, they are particle, and by the Newton's law, every particle attracts each other. So every particle attracts each other. So because of this, there is a potential energy. So there is negative potential energy between the molecules, and because of the when electron is revolving around the neutron and proton, it has also kinetic energy. So when we go to calculate, the potential energy higher than kinetic energy. That's because it is become negative. And when we calculate the total mass, it become less than if we calculate in different at a different particle.
Okay, we generally call it mass defect.
Yes, sir, mass defect.
Why there is mass defect?
Sir, because according to Einstein, the energy is also a form of actually not a form. It is actually, if we calculate mass, we have to also calculate energy, because then we will be able to calculate total mass of any system or anything, any particle. So that's why it is different. And what we calculate in normally, we see a mass and subparticles, and we calculate the mass of subparticles and we get total mass. What Einstein says, it's not true, because we also have to calculate the energy which is in the particles.
And this mass defect is associated with nuclear binding energy.
Yes, sir.
Is it true or not?
Actually, I don't know about it, sir.
Okay, okay.
There is an example which I can explain you that if we take a clock and another clock and we start the one clock, the clock when moving when clock is when the string of the clock is moving, it has some kinetic energy. The springs which are moving, it has also the kinetic energy, potential energy, and by the friction we can see a very little amount of heat can produce in the system. So that if we compute all this in the formula m is equal to E by c square, then it gives us a very small amount of mass. So if we add it in our actual mass with then it's give a very little amount of extra mass.
Okay, okay. Thank you, Ansai.
Thank you, sir.
No questions, sir.
Okay, madam.
Please proceed.
Alak, madam.
Okay, thank you, Adnan.
Thank you, ma'am.
Yeah, so the last presentation of the day is Shubham is there, the discovery of Grignard reagent. I don't see him. Shubham, are you there?
Discovery of Grignard reagent. I think he is absent.
I think he is not there. So we can conclude the session. Before that, I just request each one of you, please switch on your camera so that we can take a picture. Students, you can also switch on your cameras. Thank you so much. Yeah, it is taken now. Oh,
Thank
one
you,
more.
Gaurav.
Yeah. So I would like to thank our honorable judges who have given their valuable time, and our participants who have given so much hard work to present the all classical paper, and they have given their best. And I hope you will keep on doing this work in future also. And I would like to thank Kiran sir also for his support and his guidance. And with this, I will conclude the session. And once again, thank you, Dubey sir and Neeta madam.
Thank you,
Joining.
Thank you, Palak, madam. Thank you, Kiran sir.
For constant
Madam, thank you.
doing work at that particular end, and it's very good. Thanks a lot. And this time, I think it was much better than the previous time.
Yeah, with the time, students are also, they are learning because they listen others, and they are also learning. This is the second year we are continuing this session, Anusandhan. So, and I can see the lot of improvement in their presentation and their communication skills.
Yes.
Yeah.
Correct.
The communication skill is very important. Because
Yeah.
in physics, there were excellent papers not communicated properly, but there were many few chemistry papers were very simple but communicated in such a nice manner, I would say.
Yeah, and some culprit is our internet quality also. I would say, but no worries. In the coming year, we will be meeting face to face offline. Then it will be better, I guess.
Yes.
Then so in offline mode it will be better because no such communication gap will be there.
Yeah. So once again, thank you all of you for joining. So with this, I will end the meeting. I hope everyone has filled the attendance form.
Ma'am, can you send once, please?
Sorry.
Attendance.
Attendance form. I will. Yeah, I will.
We were giving
No worries.
presentation.
No worries. No worries. I will post one more time. You can take. You can just fill the form. Okay.
Okay, ma'am.
Yeah. Okay. Just stay there in the meeting so that you can fill. I will just.
Okay, ma'am.
Yeah.
Thank you, Palak. Thank you,
Thank-
sir. Dubey sir.
Thank you, madam.
Thank you, madam. Thank you so much.
Thank you, Kiran sir. Very nice arrangement and very nice idea about this presentation by students.
Yes, sir.
Original discovery by Nobel laureates. It's very difficult to present in few minutes
Yes.
about that discovery.
Last year, this idea was there. Choose a classic paper and present.
Correct.
But this time it is much better.
Now we can learn a lot from these papers
Yeah.
actually, if you go through in details, because they are 70 pages, 15 pages, 90 pages papers.
Yeah.
So students, you can fill the form. Yes, madam. Yes.
Thank you, ma'am.
Welcome, all of you.
Thank you, everyone. All the participants.
Yes, students, have you filled the form? Just let me know, then I can end the meeting.
Yes, ma'am.
Okay, thank you. Thank you, Dubey sir.
Thank you, madam. Thank you.
Yeah, I'll end the meeting now.
Okay.